Good morning, everyone. Actually, good morning, good evening, depends when you are. Good evening for the people from New Zealand, Australia, Asia. Good morning for people in Brazil. Midnight still in Honolulu. Okay, we're going to continue with our program. Today we will start with a presentation of Professor Elaine Martinis from the University of São Paulo. In fact, she is at University of São Paulo, but as I mentioned the first day, she is from the campus in Ribeirão, Preto, because University of São Paulo has several campuses all around the state of São Paulo, but it's still the same university. She will be presenting on taxonomy biomolecular tools and culture-dependent and culture-independent method for studying food microbial ecology, because taxonomy of the microorganism is what kind of microorganism we have in our fermented food products is really important, because we don't want pathogenic, we want only beneficial to be there. But when we have the bad guys, we need to find a way how we can reduce them and kill them. So Elaine is a professor at Faculty of Pharmaceutical Sciences of Ribeirão Preto campus of University of São Paulo, where she began her teaching career in 1998, like 26 years ago. She got a degree from University of São Paulo as a pharmacist in 1990, a master's degree after that in analytical chemistry in 93, and a PhD in food science in 97. And she was as well having the internship at Rutgers University in the US in New Jersey and University of California, Davis. She completed postdoctoral studies as well in Food and Drug Administration, CFSN, between 2005 and 2006, using a scholarship from CAPES, and later at University of Pennsylvania in 2017, using scholarship from BAP, FAPESP. So unfortunately, Elaine is not available to be with us today in presence, but she will be presenting her presentation via the video presentation. So we can see her presentation.
It's my
now.
pleasure to participate of this winter course, and I will start my presentation in a few moments, please. Well, today is the day 3, and this is the talk for taxonomy and biomolecular tools, and I will present the topic culture-dependent and culture-independent methods to studying food microbial ecology. And I thank Professor Todorov for hosting this session. My name is Elaine, Cristina Pereira de Martinis, and I come from Ribeirão Preto, a city in the southeast state of São Paulo, Brazil. And my school is the school of Ribeirão Preto School of Pharmaceutical Sciences. In this school, I have the research line, microbial ecology in food, biofilms, and interrelationships with health and disease. And to talk about culture-dependent and culture-independent methods, I chose to present some data on the case of cocoa fermentation. And this is the summary of my presentation. I divided it into 5 parts. And just before we begin, I would like to make sure about some basic principles on omics. So if you remember the central dogma of biology, you know that the DNA molecule can be transcribed to messenger rRNA, and this RNA can be translated into proteins. So when we study all the genes of a given sample or organism, we are talking about genomics. And when we do a global analysis of the rRNA, we are talking about transcriptomics. And when we study all the proteins that are synthesized by an organism, we are talking about proteomics. And we can also have metabolomics for metabolites and secretomics for peptides that are secreted from cells. So the omics, basically, they have this name because it refers to large-scale studies. So in the case of genomics, is the large-scale study of a group of genes. And the genome sequencing is the process of determining the sequence of nucleotides of genes. If you also remember the classical method of Sanger sequencing, it's the method based on the chain termination using the deoxynucleotides, and we can determine the bases that are present in the DNA in a given sequence. And high throughput sequencing methods for DNA sequencing, in fact, use the same principle of Sanger sequencing, and we have 4 types of deoxynucleotides that are labeled with fluorophores. And this kind of fluorescent labeled deoxynucleotides, they are incorporated in the DNA molecule, and the equipment is able to make reads of the nucleotides that were incorporated. And when we finish this process, we have millions of sequences that have around 100 base pairs. And so we then have to use bioinformatics to analyze these sequences against reference strings or against reference sequence. So we have many equipment that today do genome sequencing. We have Illumina, which is one of the most used ones, PacBio, Nanopore, and I am sure there are some others too that can be used commercially. So the metagenome is the collection of genes and genomes of members of the microbiota, obtained by the direct analysis with high-throughput sequencing of the total genetic material from a sample. And this name comes from meta-analysis, which is a statistical tool that quantitatively correlates and summarizes genuine association, and genomics, which is the global analysis of genes. One kind of metagenomic analysis is shotgun sequencing. For this, we have DNA extraction from the sample, a library preparation that's done with random DNA fragmentation and labeling, and we then have the massive and parallel sequencing of these fragments, and we use bioinformatics to analyze and compare these DNA fragments with reference databases and annotate or assemble these pieces. On the other hand, metataxonomics is high-throughput sequencing done to characterize all the microbiota and to create a metataxonomic tree that shows the correlation of all the sequences obtained. And for this, we, instead of doing the fragmentation, direct fragmentation of the DNA, we do an amplification of phylogenetic markers. Usually, we use 16S rRNA for bacteria and 16S rRNA for, or ITS for fungi. So, also to remember, prokaryotic cells, they have these subunits of rRNA, and so 16S is one that's very useful for taxonomic studies. And for eukaryotic cells, usually we have 16S that's used for taxonomic purpose, or we also have the internal transcribed sequences that are so between these rDNA 16S or 28S. So when we want to do taxonomical studies with fungi, ITS is a genomic region that's very useful. And when we sequence the 16S rRNA gene, we may use several variable regions, but the most commonly used are the V3 and V4 region. And this is what is sequenced when we do meta-taxonomics. That's V3 and V4. We have a PCR amplification. Then we add some index to this pieces of DNA that were amplified, and then we do the sequencing. Another approach that's very useful is the whole genome sequencing of isolates. So when we combine the study of the whole DNA that's present in a sample, using meta-taxonomics or shotgun metagenomics, we have the just a moment, please. I'll check that my recording is in progress. Just a moment. So, when we do this approach with the that we do not culture cells, it's very useful to have the metagenomics and metataxonomics, but it's even better when we combine the metataxonomics with the whole genome sequencing of isolates. So it's very useful to have also isolates. We can combine and have more information about the sample. So the first part of my talk on the case of cocoa fermentation is to answer a question: does quorum sensing play a role in microbial shifts along spontaneous fermentation of cocoa beans? And we used an in silico perspective to do that. I don't know if you are all familiar with production of chocolate, but the production of chocolate starts with the harvest of cocoa and cocoa pods, and then we have inside the cocoa beans, and the cocoa beans go through spontaneous fermentation. Then the beans are sun-dried, put in large bags, and transported to the factory. The beans are roasted, crushed, and then they are used to prepare chocolate. So this spontaneous fermentation is a microbial succession, and we have in the beginning, we have high counts of yeasts and lactic acid bacteria. The yeasts then decrease in number, lactic acid bacteria decrease slightly, and we have a rise in acetic acid bacteria. And you can check here that all this process takes around 6 days. And when we have this microbial succession, we also have changes in the concentration of different metabolites. Fructose and glucose, they are used, and the yeasts start producing ethanol, and by the end we have lactic acid, acetic acid, and also mannitol. So we published a paper in Food Research International where we were trying to answer the question if quorum sensing is involved in this process of spontaneous fermentation of cocoa. For those of you who are not familiar, I'll just say a few words about quorum sensing, which is a mechanism by which bacteria regulate gene expression in accordance with population density through the use of signal molecules. During the reproductive cycle, individual bacterium synthesize autoinducers, and these autoinducers, they vary depending if it's a gram-positive or a gram-negative bacteria. And when these autoinducers, they increase and reach a critical mass, they bind to receptors in the cells, and they trigger signaling cascades. And this may lead to alteration of transcription factor activity and gene expression. And this alteration is very important to regulate phenotypes like biofilm formation, bioluminescence, toxin production. So this kind of communication among the cells is very important to allow for the cells to adapt to different environments. So we had the hypothesis that quorum sensing was involved in the microbial succession of cocoa fermentation, and it was important for the adaptation of these strains to the environmental conditions during the fermentation. And here we can see there is a central molecule that's involved in quorum sensing, which is LUX-S, and it's an intermediate that's very important for the synthesis of autoinducers. So in this paper, we wanted to determine the microbial diversity along 6 days of spontaneous fermentation, to determine the microorganisms that would possibly be related to quorum sensing, and to determine putative effectors related to quorum sensing. And for this, we took some samples from Bahia State in the northeast of Brazil. And so I had a PhD student who was Otávio Almeida, who did this fermentation there in Bahia. And then with CEPLAC, and then we did samplings at different time points, and we did both culture, and we also saved some samples for metagenomic analysis. So we did isolation of fungi and from also bacteria. Besides the isolation, we did DNA sequencing of the whole DNA that was obtained from the fermenting beans. And then we followed the bioinformatics workflow to find what what was the microbial diversity in this sample. And for this, we used many bioinformatic pipelines to check the quality of the reads and to trim the reads that were of best quality. And we also compared this with reference data banks. So we used for this the metagenomics, the shotgun metagenomics, and we had fragments that were obtained from the samples, DNA fragments that were sequenced. And this way, we were able to check for populations of bacteria and fungi. So just showing a bit more in detail, we did sampling of the cocoa nuts in plastic bags, and they were used either for isolation of bacteria and fungi, or they were stored under frozen storage. Bioinformatic analysis, as I mentioned before, with different pipelines. And then the results. We observed that were yeasts in all phases of fermentation, and we were able to reach the determination of the at genus level. So we had many yeasts genera like Saccharomyces, Saccharomyces, Pichia, and some others that are not so common. So, and also we had lactic acid bacteria and acetic acid bacteria in the late phase of fermentation. We have bacteria from the genus Lactobacillus, also Acetobacter, Gluconobacter, Comagatae, and Enterobacter, some other genera. And we also prepared a cladogram showing the species level reconstruction from specific marker genes dispersed on cocoa metagenome, considering the entire microbiota. And we represent here those that had at least 25% of relative abundance. And so here we can see we had Acetobacter, Bacillus, Fusarium, Gluconobacter, Klebsiella, Lactobacillus. So this data, they are very important to show that the metagenomic approach can reveal a very high diversity, microbial diversity in the sample. Also, with these metagenomic studies, it's possible to look for functional genes that are important for different metabolic pathways. And we have here, along the fermentation times here, 0 to 144 hours, the distribution of different metabolic pathways. Carbohydrate metabolism, metabolism of amino acids, metabolism of terpenoids and polyketides that are very important for flavor. And so we can look into detail what are the main functional pathways that are encoded by the genome. And our question about the quorum sensing. What we could see analyzing the genomic data was that there were many quorum sensing effectors that were present in the genomes of those bacteria. And we found genes that encoded for acylase, lactonase, regulator looks as, do you remember, that central regulator for quorum sensing and transporter. And here we can also see those effectors in relation to the different genera that were present. So Acetobacter produced lots of acylase, Bacillus produced acylase, lactonase, looks as regulator, transporter. Actually did not produce, they have potential to produce, because we just analyzed the genome. We did not check if those effectors were really synthesized. We just checked the genome, okay? So the second part of our work was the analysis of pangenome. And we were looking for looks as coding genes and the enzymatic repertoire in cocoa-related lactic acid bacteria. Do you remember we did isolation studies? So we got some lactic acid bacteria, and we did the whole genome sequencing for those isolates. And we were trying to check if those isolates had the potential for quorum sensing. Because if you remember, we saw there were many quorum sensing effectors in the genome of the whole cocoa sample, like the DNA that was extracted directly from the cocoa fermenting seeds. And we now have the isolates, and we want to check in the genome of an isolate if they have this potential to produce lux S. So this work was done in collaboration with Italy, Professor Giovanna Felis and Nicola Vitullo. And the highlights for this work is that it was the first whole genome sequencing of Pediococcus acidilactice from cocoa fermentation, the first on the prevalence of lux S gene in lactic acid bacteria. And also we developed species and clade-specific primers that could be used in the future to assess lux S genes in lactic acid bacteria. I will show briefly this work, but what we did was to study 3 different strains of lactic acid bacteria that were obtained from cocoa fermentation, and we did comparative and functional analysis. And for that, we downloaded all the publicly available genomes for each species from NCBI, and we run pangenome analysis also of HGT, horizontal gene transfer. We did functional annotation and prime design for future studies. Here we show some results. We analyzed the G plus C percentual distribution of collate filtered assemblies of the lactic acid bacteria. And so we have here a comparison with 409 strains of the literature for Lactobacillus plantarum and 9 strains for Lactobacilli. And here we show the average nucleotide identity, and we confirmed the identity of our strains. And also we saw that our strains were clustered with the same type strains from the literature. And we also saw in these cocoa-related strains that they had open pangenomes. It was a genome that was very plastic. what does that heteroplasticity? What does that mean? We have a few genes that are considered core genes, and we have a high number of genes that are not present in all the strains for that species. So what this indicates, that possibly we have horizontal gene transfers. So the strains are able to adapt to different environmental conditions. And also, we observed that for these strains, we had looks as in multiple copies, which indicates that codon sensing might be active for these species, and they might be able to do horizontal gene transfer, and it's possible that this communication among the cells is important in this process. So the third part of the work on cocoa fermentation, we did a comparative pangenomic analysis and analyzed also the biotechnological potential of cocoa-related Acetobacter senegalensis strains, so acetic acid bacteria. For this, we selected the strains and did whole genome sequencing. And then we analyzed the genomes that we obtained against public genomes that were present in databases. And we did comparative analysis also to find about possible genes related to codon sensing and also to adaptation to stress conditions. And what we saw with the pangenome analysis was that there were Acetobacter senegalensis strains and with many different metabolic pathways related to stress adaptation. They were able to adapt to many stresses like oxidative stress, osmotic stress, and so depending on the strains, they have more or less of those pathways. Also, we observed the presence of genetic elements related to codon sensing to know acylmucin lactone flux protein, lactonases, and acylases. Lactonases and acylases are enzymes that might degrade codon sensing molecules. So, Acetobacter senegalensis might interfere with the codon sensing molecules produced by other strains. Also, very important, we observed there were no virulence genes or resistance to antimicrobials. It's very important because if we want to propose a technological use for those strains, they might not present any virulence genes, and it's highly desirable that they do not present resistance to antimicrobials. Overall, we observed there were good traits for strains intended to be applied in food fermentation. The fourth part of my talk is about metagenome assembled genomes to characterize microbial diversity in cocoa fermentation. For this work, we did assembling of genomes from metagenomes, and we showed it can contribute to review the microbial diversity. For that, we compared our data with data that were previously obtained by other Brazilian researchers and also some researchers from Ghana. So we compared our data with the literature. We downloaded those data from public databases and compared. And what we could see that metagenome assembled genomes can show neglected microbial diversity. We also, here we have 17 high quality samples, and we have here the microbial diversity, but we also had some medium quality megs that were recovered from cocoa microbiomes, and some of those species, they had not been previously detected in cocoa environment, like Licorylactobacillus vini, Comagataebacter saccharivorans, and Comagataebacter maltaceti. So, when we use the metagenomic data to try to assemble genomes, whole genomes, we can also take advantage of this technique to further gain knowledge on the microbial diversity. And here, for the megs, we can also evaluate what are the main metabolic pathways that are present, and according to the different species that were determined. So phenotypic traits determined with tritar pipeline showed no correlation for any trait and particular sampling sites. All lactic acid bacteria presented a set of genes for metabolism of carbohydrates, and all but one Lactobacillus fermentum harbor genes for fermenting the mannitol. Selected megs presented the ability to ferment salicin and citrate. And moreover, there were megs of lactic acid bacteria with undesirable predicted phenotypes, such as production of hydrogen sulfide and putrescine. And the functional notation done via HASTK server showed that megs of lab presented the classical lactic acid fermentative pathway, and some species also presented the potential to metabolize lactose, galactose, and other carbohydrates. So why this is important? For example, if I wanted to propose a culture media to isolate some specific strains, or not strains, but some species, I can find, for example, some carbohydrates that are used by those species and add this to a culture media, for example, if I want to do isolation. Or if I want to produce some biotechnological, some compounds of biotechnological interest, I can check which are the main species that are related to the metabolism of that specific carbohydrate. So there are many applications that can be envisioned from this data. And the part 5 of my talk, the case of cocoa fermentation, investigating interspecific quorum sensing influence on cocoa fermentation quality through defined microbial cocktails. When we started this work with that quorum sensing paper, we wanted, by the end, to have a proposal for a starting culture to be used in cocoa fermentation. Because when you have the spontaneous fermentation, you might have very good quality chocolate, but you also may end up with bad quality chocolate because you depend on the autochthonous microbiota that's present. So we wanted to check if it was possible to prepare, define the microbial cocktail and do the fermentation process. So for that, we used our isolates to do lab scale cocoa fermentation. So investigating looks as genes expression in lactobacilli along lab scale cocoa fermentation. For this, we proposed 3 conditions for a pilot scale fermentation, and we did one fermentation adding Pichia, 2 strains of Pichia and Saccharomyces. The other set of experiments with Lactobacillus plantarum and Fermentum, and a third one with Acetobacter. And so we used defined cocktails to check if we really needed all the 2 kinds of bacteria, actually, the acetic acid bacteria and lactic acid bacteria, plus the yeast to carry out the fermentation. And so we carried out these different fermentations, inoculated with the yeast, lactic acid bacteria, and or the acetic acid bacteria. So we did sampling, and we did enumeration, meta-taxonomics, we determined enzymatic activities, and we determined also volatile metabolites. And the quantitative RT-PCR for LUX-S was done to check if the quorum sensing pathways were working. So for this, we used the primers that we developed in the previous paper that I mentioned. So we used the primers to look for LUX-S, and to check this, we used the quantitative RT-PCR. So we checked the diversity, and we also checked the amount of LUX-S genes that was expressed during the fermentation. Here we have some data for this work, and we were able to follow all the species that were present in this fermentation. And also here we have the bacteria, and we also followed it for the yeasts. And we did determination of the enzymes that were present in the fermentation. You can see here that we checked the whole set of enzymes, and basically what we observed for enzymes was that plant hydrolases, they were crucial for the enzymatic profile observed. So the enzymes that came from the cocoa beans themselves were very important for the fermentation, and not only the microbial enzymes. And here I show that we observed that Lactobacillus plantarum presented the higher potential for quorum sensing. And we observed that because we were checking the expression of LUX-S gene using the quantitative RT-PCR. So we could link the high production of these Luxet molecules to Lactobacillus plantarum in different sets of fermentations. And also, what we checked, excuse me, was the influence of quorum sensing on the quality of fermentation. And for that, we did a correlation analysis of Lux-S expression and volatile compounds. And so we checked what kind of flavor would be more strong in different fermentations, and what could be the microorganisms that were involved in this expression. And what we observed was that Lactobacillus plantarum could be associated to the formation of some undesirable compounds, flavor compounds. So the conclusions for this paper were that our data corroborate former shotgun metagenomic analysis by demonstrating the expression of Lux-S by lactobacilli along cocoa fermentation. Moreover, our results indicate that Lactobacillus plantarum is the main lactic acid bacteria related to expression of Lux-S, which is a compound that's related to quorum sensing. So, what are the take-home lessons for this topic? First, cultivation techniques are still essential to understand food microbial ecology. Second, the omics tools allow to assess novel microbial taxa in food systems and to understand their taxonomy, gene expression, and metabolism. Also, the omics data should be combined with culture of microorganisms whenever possible to validate properties predicted by in silico studies. And fourth, overall, these advances should result in the better safety and quality of our food supply. So every time we can combine culture dependent and culture independent methods to evaluate the microbiota of food, this is the best, because we can do some predictions in silico, and then we have the strains to validate those findings in the food system. And this is much better to make sure our results may help in food safety and food quality. So, to finish, I want to acknowledge my current laboratory team, and also I want again to acknowledge my colleagues, Professor Chiquindas, Professor Todorov, and Professor Wellington Pinto, as well the PhD student Gabriela Santos, for working with me in this Alcani course. And finally, I want to thank you and for your attention, and say that I am also very grateful to all my collaborators in Brazil and abroad. And our work would not be possible without the funding from FAPESP, CNPq, and CAPES. So thank you very much. And I leave here my email if you want to send questions to me. I will be happy to answer. So thank you so much, and I wish you all the very best. Thanks.
Thank you very much, Elaine, for your presentation. Unfortunately, this was the recorded video because Elaine was not able to take in person participation today with us. So if you have any questions to Elaine, you can send by email. We're going to provide the email in the chat system of the now on YouTube. So you can email her and ask her some what do you want to know more about her work. It's very interesting because we just need to remember that Brazil is one of the biggest producer of cacao, and exporting and quality of the cacao products is very important, and all the microorganisms involved in this process needs to be well mapped, and we need to know what they do and how they do the entire process. So next presentation will be in a few minutes, so we can have a couple of minutes of break, and at 11 o'clock we continue our winter school with presentation of Christian Hoffmann. He will be he's already with us here, and he will be presenting on his work. So at 10 o'clock we continue with Christian Hoffmann. So a few minutes break, and I hope you will be back soon for the next one. Good morning, everyone. We can continue, and you can see it's already the Professor Christian Hoffmann is here with me. He's a professor at the Department of Food and Experimental Nutrition of the Faculty of Pharmaceutical Sciences at the University of São Paulo. He has his PhD in biology from the Federal University of Goiás, and having conducted his thesis work under the guidance of Dr. Frederick Bushman at the University of Pennsylvania. So no excuse to don't have an English presentation. Like most of Brazilians say, no, I don't want to speak in English, I'm afraid. But, well, there is no excuse because he was studying in the US and having several projects with them and working in the close collaboration. He's associate researcher at FORC, Food Research Center, and a collaborator researcher at the Applied Immunology Laboratory at the Biology Institute of the University of Brasilia. He worked for 10 years as a researcher department of the Microbiology University of Pennsylvania School of Medicine, visiting researcher for 1 year at Helmholtz Institute in Munich in Germany, experience in molecular ecology studies in various microbial systems, including soils, microbials, mats, and in the last 10 years, he has the focus on the research in the human microbiome and other hosts. As well, he's a passion for the wine and cheese and doing for his pleasure a study in these food matrices. His research interests include interaction between microbe functions in health and disease and how it modulates by diet and the immune system. So a very big pleasure to have you with us today because it's you're one of the reference in biomolecular studies at our laboratory. So really, I'm looking for your presentation. Thank you very much to be with us. And now it's you.
Thanks, Levi. Hi, everyone. Good morning, good afternoon, good evening for whoever is watching us elsewhere. So let me just test my slides here. Yes. Okay. Oh, maybe not so much. Well, we'll manage. Thanks for the introduction, Slavi. I am professor here at the School of Pharmaceutical Sciences, and my main focus is actually the human microbiome interaction. But I do like fermented systems because they actually, allow us to reduce what we can actually study much more complex systems, and then apply methods, develop methods, and then bring them into a more complex system that actually has the host, which is also living. So I do like to work with food systems as well for those reasons. The study of fermented foods is first and foremost a study of an ecological system, right? So we're talking about microbiomes. Most people tend to use the word microbiota and microbiomes as exchanged, between the two of them. They are slightly different. Microbiota really refers to the species themselves, right? What are the bugs that are within the sample that you were working with? Whereas the microbiome actually is talking about the entire system. It talks about the genes, the expression of those genes, the metabolites that actually you have within that system. And the metagenome, which sometimes is used in exchange between those two, actually refers just to the collection of all genomes combined from the system. So you will extend that as well to metatranscriptome, the collection of all the transcripts from all the different genomes that you have in the system, in a meta-analysis sort of way. And of course, we're talking about, depending on the system, from all domains of life, bacteria, archaea, virus, eukaryotes, and that, of course, depends on what you're working with. There are some main characteristics that we need to take into consideration when we're working with this: species abundance, cell density, species richness, and we will talk a little bit about some of those concepts as we talk about the methods. This talk will be very much focused in understanding the fundamentals of actually working with those systems and how we actually understand them. So just to refresh everybody's mind, ecological systems for the microorganisms are not new. We know them for a very long time. The very first description of a microbial ecosystem actually comes from, my slides did get reformatted for the StreamYard, so let's see how things will progress. The very first description of this actually comes all the way back in the 1700s. We see microbial mats actually being described as soon as we have access to microscopes. So we see complex communities of different microorganisms starting to be described as soon as we can see them. And from that moment on, we try to understand them as a system, working to actually live together and use the energy from that system. So in the case of a microbial mat that lives within, a water system, usually you have photosynthesis, and that is the primary source of carbon that actually is, being fixed within the system. In a fermented food system, you're actually adding this from somewhere, right? We're adding it as a carbohydrate coming from somewhere, or maybe protein, or maybe fat, depending on the main energy source that comes within the system. And this is not different for any one of the systems. We should understand that we're trying to understand the flow of energy within the system. As an example here, this is a simple schematic representation that actually comes from some people's guts, where we can see the three main domains of life represented. So we have fungi, shown here as Candida, we have bacteria, Rhodopella, Rhodinococcus, and we have Archaea, the Methanobrevibacter here. They all actually are working together to actually best use the energy source that's available. In this case, it is very much driven by carbohydrates. So whether those are leftover materials that actually arrive there from digestion, in this case, they will be somewhat processed by some species. The leftover from those species will be passed on to the next species, and so on, until we actually end up with CO2 or methane in the case of the Archaea, which are end products, and we usually don't find them in most systems, although there are some microbial systems that can actually use methane and CO2 further in their metabolism. But usually they are endpoint from this process of actually obtaining energy. If you understand that you're following the energy, you will probably make sense of what is happening within your system, right? And who is using what will actually help you guide your decisions in how to interpret your data and how to actually eventually manipulate that data if you actually want to improve that data. Fermented foods are not different. They are also complex communities, albeit not so complex as the gut microbial ecosystem, which is also not as complex as the soil microbial ecosystem, and so on. But we also have the same relationship of all those different organisms living together. We have, this vast array of different species doing different types of metabolisms, which will be perceived by us as consumers of that product as a sensory input. So for the bacteria, this doesn't matter. This is for us as a person that actually eats this food. But for the bacteria, they're just growing together and trying to best use that system. Whenever you break those relationships that are established, we usually perceive this as a defect because it's not what we expected. But that doesn't mean that that's actually wrong for the bacteria. This is what is right for them at that moment. So the idea of the fermented food and trying to control the system is try to make the bacteria work for us in a way that we like. But they have their own ways of doing their things, which is the best for that community at that moment. And I think that part of the understanding of relationships is really, one of the advantages of actually working with molecular methods. You don't need the microorganisms necessarily to actually try to start understanding the system. All of this came to light based on DNA sequencing. This was, of course, a Nobel Prize winning discovery all the way back in the late 70s. The Nobel Prize was awarded in 1980. And the idea actually got adapted to the work of ecology very, very soon after the method was discovered. What happened from there to today is that we changed the scale of how we work with those things. So whereas in the beginning, we actually had Sanger sequencing, which is the one that actually became commercially available. There were two different types of sequencing methods actually that shared the Nobel Prize, and one of them became commercially available. Lucky the person that actually got it, the royalties for that. But for that, we see here in the left, the type of sequencer that was commercially available. That's what sequenced the human genome, rooms and rooms full of those sequencers, as we see here. And the idea is that we had, in the early 2000s, a shift in how we did sequencing, the scale of sequencing. Just to give an example, probably the one that first really revolutionized access to scale of sequencing was the 4x4 platform. It basically could produce 1 million reads in less than 1 week, whereas for you to actually do the same kind of throughput, 1 million reads, it would take over 200 days running 24/7 to actually do that in the old style sequencer, right? So that really changed how we actually deal with the technology, and that opens up a lot of different ideas for us. This technology with 454 is retired. We don't even have it in the market anymore. This was probably around 2008 was the peak of use. And from there on, we see the expansion of all those families of sequencers to many different types of throughputs and different types of sequencers that are available today. The ones that de facto are most used are produced by Illumina. Everybody probably has heard from the name Illumina when they're talking about sequencing. And there are many different scales of work that we actually can choose from. There are other sequencer platforms available, the Nanopore for long read, more low throughput, as well as the PacBio sequencers are also available. They're considered third generation sequencing, and this Illumina scale is actually second generation sequencing, we could call it. And normally we would work with the MySeq platform within the laboratory scale for most people. We probably would work within this range of 25 million reads per run. So with 25 million sequences, you can do quite a bit of analysis. You can do quite a bit of work. And having access to that really changes how we can ask questions and how we can answer those questions. But with that said, we have a full range of different scales that you can choose from. The most up to date is the NovaSeq X, which can produce as much as 52 billion reads in a single run. So we are able to produce 48 genomes, human genomes in maybe 3 days today. So it's really an impressive amount of data that we can actually obtain. To speak for our Brazilian colleagues here, all those platforms are available in Brazil. They are quite available everywhere in the world, especially the MySeq, which is reasonably cheap within the great scale of things. The cost of the reactions, the cost of sample production is much cheaper because you can really work with the scale of the analysis, and there is possibilities of using smaller scales as well. So it's not outside of the reach of normal projects to actually work with. You can actually budget to work with those types of sequencing technologies today. The part that's really more complicated comes afterwards, right? Just to remind everyone that we don't have just Illumina. We have, for example, the third generation of sequencing, which is really shifting how we think about real-time DNA sequencing. The sequencer is actually right on screen. We can see it there being pipetted on. It's plugged directly onto the computer, and the analysis is real-time for the Oxford Nanopore sequencer. So for that, it's not something that we would probably do any type of sequencing for metagenomics, but we would do it for genomics, for sure. We would extract the DNA from the bacteria and sequence it directly onto that platform. So we would be able to access what are the genes that actually have been worked here very quickly, within a couple of days. If you're doing transcriptomics, within a couple of days, you would know which genes were active when you did an intervention in your sample. The price of the samples are also accessible. The biggest difficulty that I find of working with Nanopore, it's not even the quality of the reads, which, depending on what you're doing, could be a little bit lower, and it's fair to say that. It's not as high as, the sequence quality is not as high as we get with Illumina. But we do get, compensated in the length of the reads. The biggest difficulty is that you have to budget your time very carefully because the shelf life of the Oxford Nanopore reagents is much shorter than Illumina. So it requires very careful planning. With that said, we do that all the time. So it's just a matter of adapting how you work. What are the strategies that you actually are working with? Consider that you can obtain this data and work with data like that in real time, within your week, within your month of work, right? All the current technology that we have is on the screen. right now. Even in some places, they're still offering 4x4 sequencers, but first, second, and third generation do have their place within the biomolecular, array of tools that we have to work with. All of them have the purpose. All of them can offer us something to do, and all of them are still very much used today to answer questions. I'll present to you today mostly data that's around, or how to obtain the data that's around Illumina sequencing, because it is the most widely used platform, and it's probably the most versatile for most people. So we'll follow through with Illumina-based approaches here. But all of the ideas, they do translate to basically every single type of technology that we have. It's just a matter of really designing your experiment. So as I said, with so much data, you actually change how you collect and how you analyze the data, and what are the questions that you can actually work with. So you don't have to think that you only can do 10 replicates. You could do 100. You could have different batches. You could do different seasons throughout the year. You could do many different types of data collection and run them all into a single run of sequence acquisition, and then finish the analysis post facto. So you can change how you think about your questions in a much broader scale. And this is something that we couldn't do before because it was just cost prohibitive. Today, it is not so cost prohibitive. We can actually work with. There are kits today to do the library prep, as we call it. So once you have your DNA, you can use commercially available kits to prepare the sequencing reactions and send it to the sequencer. So you do not have to develop those reagents yourself. They are published protocols that can actually be used. If you're going to do this routinely, I would recommend that you implement protocols as opposed to use the kits. The kits are much more expensive than implementing protocols in your lab. There is the Earth Microbiome Protocol, which is very much vetted. The primers are very much vetted for all systems, and it's much, much, much cheaper to work than the Illumina kits. So if you're going to do this occasionally, consider the commercial kit for the facility of having to use it once a year. Okay, that's fine. If you're going to do this all the time, consider that then the expense will be much higher. So you could reduce those costs by implementing those methods within your lab. The data itself has two major varieties. So we have amplicon-based data and we have shotgun-based data. So the amplicon-based data is all PCR-based. So if you can do PCR, you probably can prepare samples to be sequenced with Illumina. It's as simple as that. Okay, it's a PCR reaction, and you just have to follow a protocol that is specific for this PCR reaction. So this is not that difficult. This is something that pretty much every single lab that works with molecular biology could do. It's not outside of what we have been doing for the last 30 years. So it's not that complicated. The question then becomes, what is it that I am actually amplifying? For bacteria, normally we amplify the 16S gene, right? So the ribosome is this universal product that all living cells have. And we do have primers that actually work really well for bacterial RNA genes. But we could use many different types of amplicons. We can do ITS regions for fungal sequences or other eukaryotic sequences. We can do 18S as well for eukaryotic cells. We can do specific viral regions. Viruses do not have such widespread genes that actually we can actually sequence all viruses with a single gene. But if you have questions that are specific, group specific, you could still use primers that actually amplify those groups specifically. So you can do PCR approach here. And you can do this for any type of gene that you would like to sequence in parallel. The idea is that you're raising data in parallel from all the bacteria, from all the samples at the same time. And then on the bioinformatic pipeline, you're separating them. Okay? The shotgun approach is what we would think as the full metagenome sequencing. Here, we're really sequencing all the genomes from all the species that are present within your sample. And of course, the more you sequence, the better you are to actually see things that are rare. So think about it this way. If you have a phage in your system, it's very small. It's a very, very small piece of DNA. And you have hundreds of thousands of copies of a genome from a bacteria. Even if you have hundreds of thousands of copies of that phage, it will still be much, much, much less DNA for the phage than it is for the bacteria. So if you were going to extract it all together and sequence it all together, it would be much harder to see the DNA from the phage. So the amount of those different species within the system actually is something to consider if you actually are thinking about the rare groups. If you're thinking about the abundant groups, that's not so much of a concern, right? It would be very easy to see everything of a bacteria that's abundant within your system. So those are some considerations that we have to start thinking when we actually deal with this type of data. The pipeline of work is actually represented here for a human microbiome, but you can substitute the microbiome for any microbiome. You have those two major approaches. You can do, a PCR-based approach for 16S, for ITS, and then you sequence that marker gene that actually can be a representative of the species that you have present, or you can do full shotgun sequencing, all the sequences that actually were obtained from that sample simultaneously. And here we have the opportunity then to do metagenomic sequencing. We're working really with the genome, DNA. We can do the metatranscriptomic sequencing. We extracted the RNA from the samples, and then we're sequencing RNA that express genes from this system. But we could also, for example, separate the viral particles if you're interested in the phage within the system. In this case, it's really recommended that you separate the phage particles first from all the other cells, because, again, much smaller amounts in terms of quantities of the nucleic acid. So you would like to see them, and you have to separate the ones that actually are much more abundant. And for that, usually it's filtration method-based, filtration-based methods that we have available, or you can do gradient centrifugation. There are very standard methods that are used for well over 50 years now. The filtration ones are kits that we actually can buy. So if you have an issue in bacteriophages, for example, that's the approach that you would like to follow. And I'm sorry to say, most of the time will actually be spent doing the entire bottom half of the slide, the data analysis. So the biggest part here of time that should be budgeted is data analysis. I normally would say that if you're planning a project, you should plan no less than 50% of the total time of that project to be in data analysis. And why is that? The reason is because once you have the data, new questions will appear. And you have to have the time to really talk with the literature and look at the data that's already in the databases and compare it to the databases and bring new questions and interpret the data. The pipeline itself of just saying which sequence it is, today is a standard. I mean, we have standard tools that we can use to actually do the classification of sequence. We can do BLAST very easily for a very long time. So why not use the tools that we already have? They exist, they're standard. But to actually make sense of the data requires time. And the more complex the data is, the more time you need to actually work with it. The second part that actually takes time is sample acquisition. You do not collect all the samples that you need to work with in a day. If you're going to do field work, that will take time. You have to go to the field, you have to actually sample, you have to bring it to the lab, you have to extract the DNA. So all that part of the work is longer. But extracting DNA, we can extract it with 96 sample kits. So that's not so complicated. In a single sitting, you can extract 24 samples. In a week, you could extract 100 samples very easily. So obtaining 100 samples takes much more time than it takes for you to actually process 100 samples and prepare them for sequencing. So in terms of time, the time that needs to be budgeted for you to work with is sample acquisition and data analysis. Within 1 month, you probably could finish completely all the work that you would do with preparation of samples for an Illumina sequencing. So let's say you have 250 samples, 1 week for DNA extractions, 1 week for library preps, 1 week for sequencing. By the end of 1 month, you will have your sequences. That's not that complicated. Maybe things happened and you had to repeat this step. 1 more week to repeat everything on that one step. So that part is not complicated because that part is standardized. PCR, easy. DNA extraction, easy. Data analysis, easy, but takes time. Easy, but takes time. We'll talk a little bit about the data analysis because it is, there are some concepts that are really important for you guys to really understand here. Let's see my time here. Yeah. So I'll give you an example of the 16S because it is the ones that you guys are probably most likely to work with initially. The ribosome is called a universal marker because for a cell to be considered living, it has to do protein synthesis. To do protein synthesis, you have to have ribosomes. If a cell doesn't have ribosomes, it dies. You're never going to see that. So the idea of this universal marker exists for many different genes, for many different groups. The 16S works for a general marker of prokaryotes and eukaryotes too, right? It's not necessarily specific for strain work. So if you're going to have to differentiate strains, you will have to choose perhaps a different gene because it doesn't have that level of resolution to separate different strains within this species. But the idea is that we have regions within the ribosomal molecule which are really variable. They're very different. They accumulate mutations with evolution. And different groups accumulate different mutations within those regions. They're the variable regions within the molecule. And we have extended primers to be able to amplify the gene for all those regions. For a very long time, actually, we have those primers. The ones that are mostly used for Illumina sequencing obtained before a region and maybe before V3, or in that middle of the molecule is usually where we're sequencing, but you'll find primers that are standard for working for every part of the molecule. The Illumina commercial kits actually work with the V4 region, so that's the recommended region right now. For ITS sequencing, for fungi, if you're working with yeasts and you would like to actually evaluate yeasts within your system, so the ITS region is a better region to actually resolve differentiation between different fungal sequences. The 18S, which is the ribosomal sequence for eukaryotes, is far too conserved sometimes for eukaryotes. So if you want a little bit more of a differentiation for groups that are more similar, you do want to work with ITS. And there are standard primers for that too. Same protocol, you just change the amplicon. It is a much more variable region. The length of it can actually be quite different for different fungi. So you may have to adapt a little bit of the methods in the bench, a little bit more complicated, but not outside of the realm of what we're working with. So for the shotgun, we have a procedure which is a little bit more complicated. We have to extract the DNA. That part is easy enough. But the part of the library prep needs to be understood in a way that's far more complex. So when you do genome sequencing, and I will start with the genome so you understand this, right? When we do the genome sequencing, we have many different copies of that one genome that we're working with. We extract the DNA from a pure culture. So you have many copies of that genome within your little tube. What we do is we break all of those genome copies randomly. So we produce fragments randomly. And then we sequence those fragments randomly. And in the computer, we put them back together. So the figure represents what we're actually doing in the analytical pipeline of putting back together the original sequence that were represented in all those copies. Now that's relatively easy when you have a purified colony. You have a pure sample to work with. In the metagenome world, you have hundreds of species simultaneously within the same tube, and we're doing this today. So there are some constraints in what you can do. Sometimes you will not be able to assemble completely the genomes. And there's an entire line of analytical tools that actually need to be used to help you separate those data. Now, this is more complicated analysis, and this will take more time than 16S work. 16S is straightforward. Metagenomes, full metagenomes for a shotgun, a little bit more complicated. In a visual representation, usually what we would like to happen is what we have here on the left. So we have different genomes from different species, and once I sequence everything, I'll reassemble those genomes, and everything will be hunky-dory. Unfortunately, what happens is that sometimes you have similarity between those genomes, and you end up making chimeric conflicts, for example. So by similarity, by sequence similarity, then you start to actually put together fake genomes that didn't really happen, and we don't want that. We would be very biased if we did that. So normally we tend to play safe and use constraints that prevent that. But on the other hand, then you also probably have incomplete genomes in the process, many incomplete genomes in the process. So there are just constraints that actually have to be taken under consideration when you're thinking about the data. And the same thing goes for transcriptomes. The same thing goes for a single genome, if not, it's not pure calling. The standard pipeline, just to remind everyone, then is to work in parallel. We do get all of our samples. You do field work or you do experimental work. You have many different time points, or you have many different replicates, or you have many different tests that you did to a system. And you collect all those samples, extract them all, prepare them in parallel, mix it all in your liquid, put inside of the sequencer, and then you do the bioinformatic pipeline, right? And here's where really you get the data. In the end, you would like to spread it out again and know which species was within each sample that you started with. And that's where we actually end up spending the time to actually get the data. The part that actually is the simple one is the part that's inside of the tube and inside of the tube. That part is simple. The data analysis itself has some basic concepts that I think that are important for you to remember what can and what cannot be done with the data that we actually get. So the four top choices of data that you actually will get will be output data diversity. If you're doing shotgun sequencing, you can do a metabolic function, so you can actually find out which genes are present within your system. If you're doing metatranscriptome, which genes were active in that system. Of course, you will be able to do taxonomic analysis. You will be able to identify species that are present in your system. You'll be able to actually work with that identification and to do beta and alpha diversity. And to a certain level, you'll be able to quantify some of your data. There are sequencing methods established that do actually true sequencing and quantification at the same time, but they are very specific pipelines. They're not the commercial pipelines. The commercial kits actually give you a relative quantification. So usually you work in terms of, I had 100% of species and abundance of everything inside of a tube, and those are the relationships that I have here. This is the most abundant, second most abundant, third most abundant, right? So normally we would work with data that's a relative quantification, but there are sequencing methods that actually do true quantification down to the number of molecules that were there inside of you to begin with. And they are much more involved to actually prepare the samples to do that. When you have the data on hand, then you will probably proceed on to an exploratory data analysis, data visualization. There's a lot of language-specific tools that actually we work with when we're doing microbiome analysis. And when I say language, is their presentation of data that may not be familiar to everyone. So I'll try to present a couple of them very quickly. And then, of course, nothing in science is just because it's pretty. It has to have a statistical test behind it to tell you if it's really different or not. And there are statistical tests that are very specific. And then there are those that are very, very common and very, very known to everyone. T-tests and Mann-Whitney tests. Those are common tests that we've had for a very long time. And we still use them, and they're still very powerful. And they have their place within the pipeline of analysis. And you should try to understand that there is no sequencing that will solve your problems if you have no statistical power. And this is one of the advantages of working with high-throughput sequencing, is the fact that you can actually have power. You can have the sample that needs 5, 6, 7, 8, 10 replicates, and you can do the 10 replicates. It's possible to do that. Whereas for doing this with traditional sequencing would be just so expensive, you'll never do it. So you have maybe like 2 replicates. And then the error is so big, you won't not be able to see the differences because it's a very variable system. It's a living system, right? You have an ecological system. So it could be quite variable, especially if you're working with primary samples that come from the field, from not from industry-based standardized samples, but from samples that actually are in open air, for example, that have the opportunity to actually be inoculated from the air, or that's part of the process itself. So you have to remember that there is that variability here. But with so many samples available for you, you'll be able to actually take that into consideration and resolve the differences that you actually need to have statistical power. Consider that when you're actually designing your experiments. At the end, we'll have a table of sequences. It will be basically a sequence count table that will look something like this. So we'll have many different samples, and it could be taxon, right? So we could have many different taxa as the lines. We could have ASVs, which is a bioinformatic marker of species that we use, the amplicon sequence variant, if you're doing amplicon sequencing. Today, we work with ASVs. We do not work with OTUs anymore, operational taxonomic units. ASVs are a much more reliable way of really saying this is really one group of sequences and this is another one, whereas the OTU is in a statistical approach. It just says 97% on average here, or more of similarity for this group of sequences. It could be genes, right? Transcripts or genes that were found within the metagenome. And in the end, you're going to have a table of counts. I saw 10 sequences for this genus A for the sample 1. And I only saw 1 sequence for this taxon for sample 3. Or I saw an ASV that was really abundant, really prevalent, right, on 1 sample, and it was not in another sample, and so on. With this data in hands, then you can transform this in percentages, right? That's where the relative quantification comes from. So you know percentage-wise what is that taxon within that sample. And the reason is because the sequencing method is not perfect. We do have variation in the samples, right? So there will be some samples that will be a little bit higher, a little bit lower in the amount of sequences that you obtain. There are methods that we use to normalize this. The simplest one is to transform them into proportions. There are other more advanced methods, but normally we would start with just proportions to work with. And there's a lot of stuff that you can do with the proportions. So with your little table on hand, then you have to make sense of it. So this table for 16S today has standard pipelines. The one that's mostly used is Chime. Right? It was developed quite some time ago. The database is quite good to identify 16S, so you will be able to assign a name to those sequences that you saw, to those taxa that you saw. For metagenomic sequencing, we have pipelines today that are also implemented within Chime. Chime now has 2 flavors. It has the amplicon-based Chime and the shotgun-based Chime. So there are standard tools that you can actually just install on the computer and run it basically as it is already established, standardized for you. And at the end, you're going to get a little table like that to work with. With that table is where you actually do your analysis. That's the data that you need to work with to do the analysis. With that table, then you can do quite a bit of question answering. One of them is about diversity, right? I think one of the first questions when we actually think about microbiome data analysis is that we have the opportunity to actually describe in terms of what has more species, what has less species, what's the relationship between those species. There are 3 types of diversity from ecology that we actually can identify: alpha diversity, beta diversity, and gamma diversity. Beta diversity, you guys probably cannot see here. It is the relationship between samples. So alpha diversity describes how many species and what is the relationship within a sample. Beta diversity is always a question between samples. How similar are they? How different are they? Gamma diversity is a concept that we normally would not work with microbial ecology because we're not able to determine the boundaries of what is really, what does it finish where you're studying? Where does cheese finish, right? Where does cheese finish and where does the inoculum finish? So to determine those boundaries, it's a little blurry line often. So we normally do not work with gamma diversity within the microbiology scale. But it could be worked in that way. With those taxa, we can answer. No, you cannot see. It's yellow. Yeah, this was supposed to be white. The background was supposed to be white. It's just unfortunate. But the slides, I hope they will be posted. I can make them available for you guys as a PDF, and then it'll be okay. So with the diversity analysis, then the questions are related to abundance. How dominant are some species? Do we have dominant species, or is everything very equitable? Do I have all the species more or less at the same level within the system, and this means something? Or do I have a dominant species? Does this dominance change over time? Do I have a change in this major species at that moment, and later on as a different species? So those questions can be answered with this type of data. Then it's a time series data, right? If you have that question. But that would be answered with some of those indices. You can use the phylogeny itself to help guide your questions. When you have two species that are very similar within a system, that is very different than having two species that are very, very different. Let me give you an example. If you sequence a sample and you see 3 species, and one of them is a bifidobacterium, one is lactobacillus, and one is E. coli, that is an absolute richness of 3. You saw 3 species. And then you went and you sequenced another sample, and you saw that it had lactobacillus, it had bacillus, and it had bifidobacterium. You also have a richness of 3. It's also 3. But that's very different than having an E. coli or not in the system. And the phylogeny helps you guide that information. And you can capture that information using the phylogeny, because E. coli is much, much farther away, phylogenetically speaking, to the other species than bacillus and lactobacillus, for example. So we can use the phylogeny to actually help guide those calculations. Usually that will be the case phylogeny diversity index. The standard diversity index today, we use it for quite some time. Actually, quite some time. It was actually first developed for use in archaeology. And then we proceed on to represent those samples, right? Represent that information. So just come back here to our example data here that I presented to you. Normally we would not look at the sequence count table, right? Nobody wants to search numbers. We would usually represent those numbers using colors. So the standard representation within the world of the microbiome is using heat maps. The heat maps are basically a mapping of colors for each one of those numbers. So in this case here, white is 0, and the very dark color here in purple is a very high abundance for that sample, right? So we usually would try to make sense of this using statistical methods. We could do cluster analysis, we could do different types of analysis, but we would see samples that, for example, are very similar in their profile. The idea is they have the same species with the same amounts, and they're very similar to each other. You can see them very close to each other in the heat map. So the heat map helps you see things that actually may not be very visible within the table. And normally we do not stare at the table of data at all. As far as the different types of species diversity indices that we have in the literature, there is an abundance of them. There are many, many, many different ones. Those are some of the most common ones that actually we'll be able to work with. We have Shannon Diversity Index, which is very, very commonly seen in the literature. You'll see this all the time. It does take into consideration who is there and how much you have of that species within your sample. You can use it for genes as well. It is completely independent of what it is that you're using to make the calculation. You can have a Shannon Diversity Index and all of those indices for any type of measurement. What you make with that information is an interpretation, right? What does it mean to have many different genes? What does it mean to have very uneven expression of genes, right? So this is an interpretation question. And of course, then we can add a metric that actually uses the phylogenetic analysis. Just as an example here on the bottom, I have the calculated diversity index for each one of those samples from the different indices. So you can see that some of them are quite similar in terms of the distribution of data, and they have very, very distinct Shannon index, for example, from 1 to 1.7. So it's a double, right? The diversity based on that measurement, and so on. The phylogenetic diversity in this then takes the consideration of how similar samples, the species within a sample are, how well spread out throughout the phylogenetic tree are your samples, or are they very clustered, are all very similar, right? That information then weights in your metric, and you can actually interpret this in a very, very different way. Because the data is uneven, because the data is prone to actually be different between samples, for alpha diversity metrics, you should always prefer to use a rarefaction method. Use it with care. What does this mean? What is a rarefaction procedure? The rarefaction procedure is a way of saying, if I had sequenced all my samples at the same exact level, then I have removed that variability that was completely technical of obtaining sequences. The difference of actually calculating 1.0010203 that you got from the OD reading or whatever it was that was rounded up, or the fluorescence-based quantification that you used for DNA, the different number of molecules that went into a tube for making a PCR, all of this will be slightly different, right? Depending on how you prepare your samples for sequencing, which is irrelevant really for the questions that are at hand. We do not care that there was one molecule difference in the tube of PCR versus the other tube. But this will actually have additive effects. And in the end, when you actually decode everything in the bioinformatics and you put all the sequences per sample, you have a bit of differences. You have a sample that have 2,000 sequences versus 2,500 sequences versus 3,000 sequences versus 10,000 sequences. And because there is a sampling effect within the sequencer, then the more sequences you actually read in the sequencer, the more likely you are to see more species. It's just simple numbers, right? You just sequenced more, you see more species. So for alpha diversity metrics, you should always consider rarefaction. You should always try to normalize it down to, let's say, 2,000 sequences per sample, everybody. So we randomly selected those sequences, and then we do the analysis just with those randomly selected 2,000 sequences for alpha diversity metrics. Of course, to do that, you're throwing away data, right? You're basically ignoring all the rest of the data that you left out. So it's a pro and con kind of situation. You know that you have a more normal situation. You know that you're more likely to interpret the results wrongly because you try to control the depth of the sequencing, but you also left some data out. So there could be some real differences that were left out because of this. So use it with care. Normally for alpha diversity analysis, it's accepted and it's expected that you will do this kind of normalization. But for other types of analysis, you probably don't want to use it. This is much less likely to be a problem when you're working with the proportions. They tend to be represented really well. The ones that are very rare will have a very low proportion in your sample anyway. So you're likely to filter them out. Let's say you have 100 samples and you saw a group that was 0.0001% in one sample only. We're going to do it at 0.0001% on one sample only out of 100. You have no statistical power to do any other tests with this. So this is likely to be filtered out anyway in your analysis, right? Because you're not going to be able to draw conclusions from this anyway to begin with. So in the proportion world, we tend to not worry so much about this difference of depth because you'll be filtering out things anyway. But for alpha diversity, it's something that you want to keep in mind. The beta diversity then is something that will take this information of who is there and how much you have and ask how similar is the distribution of those species between two samples. So do I have the same species and there are the same amounts exactly in two samples? There's no distance, there's no difference between those two samples in the global level. So the beta diversity is always a calculation around that. Those are some of the most standard indices that we work with for data diversity analysis. Jacquard and Bray-Curtis are the ones that are non-phylogenetic. They just care about different taxa and how much we have it. In the case of jacquard, it's presence absence. It's only a question of whether you have the same species in the two samples or you don't, right? Bray-Curtis takes under consideration the abundance of them as well. How much do I have of each one of those species? And is this abundance the same in the two samples? And their phylogenetic counterparts are weighted and unweighted unifrac. Okay. The formulas are here just as a reference for you guys to actually think about it. Just in terms of visual representation, we have here the calculations for very, very small changes within your matrix. So let me walk you through this, because this is an important concept that you guys need to take under consideration when you're doing that. So what we have on the left, the top left, is the heat map that actually represents the data that I showed you before. And I marked with the blue, the cyan boxes, only two sequence counts that I changed from the left to the right. Right? Those I changed as numbers. I removed sequences from that. So you can see that the location of those boxes on the left has changed considerably, and the representation of color has also changed considerably for those two boxes. But we also have changes that happen throughout the sample, because it is proportions of the total of sequences within that sample. So the magenta and the green boxes here on the left, it's what we had before. And the same boxes are represented on the right. And you can see the colors changing substantially for each one of those. And those changes happen not because those sequences changed. They changed because I calculated proportions. So if you take one very abundant species and it's suddenly not abundant anymore because you removed those sequences, or it was removed in one of your procedures, in a rarefaction procedure, for example, then you could change the calculations, you can change the representation, and the change in the diversity indices is on the bottom. We see the pairwise diversity differences on the bottom of the slide for the representation that's on the left and on the right. Of course, in terms of presence absence, is still the same. The species is still there. I'm still saying that the species are there. I just said it is different amount, right, between the left and the right. So there are considerations that you need to take very carefully here, that you need to consider very carefully when you're thinking about the processing of the data, if you're doing rarefaction analysis, if you're actually removing anything a priori. Right? Unifrac is the beta diversity phylogenetic metric. It works the same way that we did before. We also take into consideration the fact that we have very similar or very distinct species within the sample, and that distinction is important. Whereas with the non-phylogenetic, we don't know anything about that. It could be completely different species, but the numbers are still the same or very similar. So the calculation will put them very close to each other, right? Whereas in the phylogenetic world, we would be able to see that. We will see that the phylogenetic history of those sequences is completely distinct between the two samples. With the beta diversity work, we normally go to ordination and clustering. For all data that's univariate, like alpha diversity, we basically represented in the same way that we would represent any type of univariate measure. Box plots, means, standard deviations, right? Standard errors, depending on what you're doing. So normally we would represent them in the same way. Normally we would test them statistically in the same way. We would usually use n-Whitney or t-test, or maybe interesting correlations. We do Spearman correlations, for example. But for beta diversity, normally we will use ordination. And that usually is a principal coordinate analysis or non-metric multidimensional scaling. So those are the two types that we mostly will see on the literature for beta diversity representation. If you have 100 samples, you don't want to stare at a table that's 100 per 100 cells, 1,000 cells looking at numbers. again, you would like to see this represented visually, so you can actually interpret the data. There are many, many different types of methods for clustering. Each one has pros and cons. You need to know what are the constraints of each one of those based on your question. So formulate your question. What is it that I'm interested at this moment? Do I care about very broad similarities, or do I care about minimum similarities between samples to bring them together within the representation? And this usually will guide you in choosing a clustering method if you have enough samples for that. Just as an example for the data that I presented before. So on the left, I have here the data that was my original data that I presented to you. If we do Bray-Curtis or Jacquard diversity indices for them, and we represent them on the screen, we have them on the left. And if I do that small change on just those two samples, those two species, I can see that the representation actually changes on the bottom. So Jacquard uses presence absence only. So of course it doesn't change at all between the two of them. And we can see that two samples actually become much more similar to each other that were much farther apart in the Bray-Curtis representation. So the Bray-Curtis representation in ordination space tends to represent visually all that global difference that we see there. The interpretation is always global. It's always what are samples that are very similar to each other based on this metric that I'm working with right now. Okay, so interpretation is always about global similarity. Close to each other in the graph, they're very similar. Far away in the graph, they're much more different. Points that are very close, you can make an inference that they share something. And then is a question of what is it that they're sharing? But then that's a different method. To test for this, like I said, we will actually go for alpha diversity or beta diversity. We will test this in different methods. The statistical methods for testing for alpha diversity, they're usually the univariate methods. So we can do ANOVA, for example, if you have two groups and you have a measure of alpha diversity. What is the difference between diversity between the two groups? Control versus treatment. Cheeses from one region of the world versus another region of the world. Are they more or less a species in each one of those different systems? For the beta diversity, we will have multivariate data and we'll have different types of tests. The Permanova test is the one that you usually would work with if you have groups and you would like to ask if the distances are different between the two groups. The idea is the same as we would have with ANOVA. The test itself is different, but the idea is the same and the interpretation is the same. So the idea is that you have two groups of samples and you have difference or not between those two groups of samples. And they're interpreted in the same way that you do with ANOVA. If you're not sure about the normality, about this data being normally distributed, just use the non-parametric versions of that. Ment-Whitney and Friedman and Kruskal-Wallis for this data. For the proportion data, use the same type of statistics. They're just a measurement of that species. Now you have a single species and you measured it in 100 samples. That's a simple statistic around this data. So you'll be able to work with univariate analysis for that kind of data as well. I was going to go into the details of the Permanova, but for the sake of time, I will not. It is, suffice to say, it is in the slides for you guys to be able to see. The idea works in the same way as the ANOVA actually works. It's the only test that's non-standard statistic based on what you are likely to have seen before. And usually it's a permutation-based kind of test. So I will not go into this, but suffice to say that it is a specific test that we use to test against beta diversity. So I'll finish just by pointing a couple of presentations that are in the literature, which I think are very pretty. And I think one of the things about working with microbiome data is that the data is usually pretty. We look at the graphs and we think it's very pretty. But I'll present it to you just some ideas from the very simple to the very complex of what we can actually do. So here we have just the characterization. This paper is in the literature for many years now. It was a very large characterization of many different types of cheeses both by 16S sequencing and by ITS sequencing. And what we have, the colors actually represent a different bacteria and different fungi. On the top, bacteria, on the bottom, they're fungi, and those are just bar graphs. They're stacked bar graphs for each one of the samples. So we can see how similar some of those samples are to each other. Then the clustering that we see on the top basically takes under consideration those proportions of microorganisms and pull them together in the graph to actually show what is very similar to each other, being close to each other in the graph. So this works with that table of data that I presented to you, and we gave names to each one of the taxa, right? We gave the name of the bacteria. We can do that with bioinformatic methods. We gave name to the fungi that was sequenced, and then we presented them as a stacked bar graph. Okay? So simple presentation, but very powerful, and we can immediately see many different types of cheeses in this case that are very similar to each other, right? The same types of bacteria present within the same samples, right? The same paper presented also a time series analysis for batches of standardized culture that we're doing in the lab, and that dissected out, isolated the bacteria, brought it in together, mixed them together within the lab, and then made some cultures. So we have here a time series from 1, 7, and so on, the days on the x-axis, and the quantity of each one of those bacteria for each one of the batches that actually did, or fungi, over time. So we can see that the very first time point, we have this huge amount of this bacteria that's in red. The names are even not important right now. And we basically have, we do not have the blue one, right? The bacteria only appears at the very end of the time series for those batches. And the same we can see for the Candida versus the penicillium for the fungi in the bottom. So we can do time series analysis. Then with the time series data, then we can do statistics, or we can actually probe, we can actually think about who is doing what within the system at any given time, in this case, right? Simple presentation, same type of data, but obtained over time for different batches here. We have some examples, and I'll finish with this example from a brewery, which is a more complex kind of analysis, which is possible to be done. So just to give you a dimension of the power. So what they did in this study was to actually sample the brewery that we're working with. We have here each one of the areas that was actually sampled within the study. So we have floor, we have barrels, we have walls, we have sinks, we have the entire area of this brewery that was actually sampled. It was sampled in 2 different times, and those were different times in the context of the brewing production. And what they did was determine the amount of bacteria and which bacteria are present for each one of those samples at those 2 time points. And with that, we can actually map this back onto the space that actually we have. So the heat map here is actually constructed in the same way as the brewery actually is constructed. It's the same physical map. I'll go back to it so you guys see, right? So you see the brewery, the main area with the little storage area on the side. We sampled each one of those areas. And then here we can see the representation for many different bacteria. Each row here is a different bacteria that was detected at a given time point. In red, it's a lot of that bacteria at that moment in that room. And in blue, it's much less. And we can see how that actually changes from one season to the other season, in this case, from autumn to spring in the production sequence that actually used. So this is the same type of data. It is just being analyzed and used in a different way. You're thinking about spatially. For the brewery, in this case, makes sense to understand where are potential sources of microorganisms that are coming within the brewing system. Are they actually going into the batch itself, or are they just in the environment and I don't detect them in the beer, for example? So we can do that for this type of data. Same exact data, completely different question, completely different interpretation, and both of them possible with the same type of data. This is what I had to present for you guys today. It was basically a very general overview of the potential of how to obtain the data what are the caveats of obtaining this data? What do I need to think about when I actually am designing, when I design the experiment? And to understand that you have the possibility to ask questions that are far more complex than we had before. You can answer them based on the data. That requires good planning. But with good planning, you could answer very, very interesting questions, right? That's it. If you guys have any questions, you can contact me via email. And thanks.
And thank you very much for the presentation. It was qualifying several questions. Unfortunately, you are a little bit late. As a typical Latin American, taking microphones, you know that you can enjoy talking, but it was nice. Sorry, but questions need to be after that by your personal email because we are a little bit out of the schedule.
Yeah. If anybody has any questions, just write me by email. I'll be happy to answer any question.
And we can put it your email in the chat system of the
go
streaming,
ahead.
So you can. They will contact you and ask.
Is anybody posting right now on the streaming, or should I post on the streaming?
No, Wellington will post.
You posted? Okay. Yeah, it's on the slides as well. I'll make them available so people will have it as a reference.
Okay. Thank you very much. And again, let's go for the next one. So next presenter, it's will be online with us in a few seconds. It's Professor Mohamed Merzoug. He's a lecturer of the High School of the Biological Science in Oran of Algeria. He holds a PhD in microbial biotechnology and process expertise in lactic acid bacteria, bacteriocin, molecular biology, and genetic engineering. He's also a the founder of the Genetics and Technology Platform at the University there in Oran. So thank you very much to be with us. You can actually upload your camera now, and we can start with your presentation.
Hello, Slavi. How are you doing? Thank you for the presentation. Are you here? You're hearing me?
Plaisir de te voir, mais je vois rien. Je vois juste le logo de ton université.
Yeah, I have a little problem with my...
Okay, sorry, but you need to speak in English, no French today. Or Portuguese if you want.
No French today. No Arabic, no French today. I can speak Portuguese. Impossible for me. So you can see my screen here.
Yes, we can.
Okay.
Just you can do it the big screen.
Okay, I'm trying to do. Is it the big screen now? Is it on presenter mode now? Can you hear me? Hello? Can you hear me? I can't hear you. You are hearing me? Can you make sign that I know you are hearing me, please? No. Okay. Thank you. Okay. I will start then. Thank you. So I will make it as Well, if you are hearing me, we start right now.
Can you try to share your presentation again and
Okay.
try to start?
I'll try again. Okay.
Okay, you can start your presentation.
You can see the presentation now. You can see the full mode presenter.
You need to start the presentation share in the big screen and then start to sharing.
Okay.
No, it's okay. You can
Yes.
talk in this way and you can change the slides. But you
Let's be a little bit. So I have to share like this and down. What about now? You can see the presentation now with
no,
animation?
not yet. Now it's coming. Yeah, now it's okay. Now you can
Yeah,
talk.
okay, thank you. Yeah, thank you. So, good evening, everyone. Welcome to today's lecture on molecular cloning and heterologous expression of genes. So, I am, as I have been presented by our friend Todorov, Mohamed Merzoug. I am a lecturer at Higher School of Biological Science of Oran in Algeria. Our native language is Arabic, and we speak English and French as well. So I'll be presenting in English today. I hold a PhD in microbial biotechnology, and my research interests encompass diverse topics within the field of molecular microbiology, including lactic acid bacteria and bacteriocins. And I am currently working on a project aiming at the valorization of biomolecules of technological interest produced by extremophilic microorganisms. So during the today lecture, we'll cover several key sections, of which is crucial to learn more about cloning. First, we'll define the cloning and tries its timeline, highlighting some key milestones. And next, we'll examine the PCR-based cloning workflow in detail, so you'll learn about the essential step from DNA amplification to vector integration and transformation. And finally, we'll discuss and try to explain more about heterologous expression of recombinant proteins. So without further ado, let's start with the cloning definition. As simple as it is, cloning is a powerful scientific tool that allows us to create genetically identical copies of organism cell or DNA fragments. There are two main types of cloning: reproductive cloning and molecular cloning. When reproductive cloning aims to produce a whole new organism that is an exact genetic replica of another. This method has captured public's imagination with examples like Dolly the sheep, the first mammal cloned from another cell. On the other hand, molecular cloning focuses on the cellular level. It involves the amplification of specific DNA fragments and then integrated into a vector, which then be introduced in microorganisms. Any double-stranded DNA fragment can be cloned and amplified in bacterial system, including cDNA, genomic DNA fragments, PCR products, and resection fragments. So it depends on what you want to actually do with the final product. So before we get into details of molecular cloning, let's explore some milestones that have marked the evolution of these techniques. The journey actually began, as you know, in 1953 with the groundbreaking discovery of the DNA double helix structure by Watson and Crick, based on Rosalind Franklin and Maurice Wilkins' works. This marked actually the dawn of molecular biology. Moving to the 1970s, the discovery of resection endonucleases paved the way for DNA manipulation. And this era saw the first recombinant DNA molecule created as well by Paul Berg in 1972. In the 1980s, Carey Mullis revolutionized DNA cloning with the polymerase chain reaction, currently known as PCR. It allows the amplification of specific DNA sequences using specific primers. The decade also witnessed the first commercial recombinant DNA product, which is the famous human insulin-named humulin produced using Ecology. Entering the 2000s and beyond, synthetic biology and CRISPR-Cas9 technologies began transforming how we edit and manipulate genes. And today, many molecular approaches are available, ranging from traditional cloning techniques to advanced genome editing tools. These approaches are being used to develop new treatments for diseases, improve agricultural crops, and also address environmental challenges. Let's move now to the heart of our lecture. Well, imagine you have discovered a gene in a rare plant that you believe could produce a valuable component, but the plant is difficult to cultivate in large quantities. Well, this is where molecular cloning comes into play. By isolating this gene and inserting it into a more manageable organism like E. coli or yeast, you can produce the component in large quantities. As you can see on this slide, molecular cloning involves taking, I'll try to use the pointer here. Excuse me, well, here it is. So molecular cloning involves taking a DNA fragment of interest from one organism and insert it into a self-replicating genetic element such as bacterial plasmid. This allows the gene to be copied and manipulated without altering its original sequence. Once we have cloned the gene, the next step is heterologous expression. So this approach involves expressing the clonal gene in different host organisms. By doing so, you can produce large amounts of your recombinant protein encoded by the target gene. So as I mentioned earlier, our focus today will be on PCR-based cloning. This technique is versatile, allowing us to clone almost any DNA fragment into a vector of choice with very few limitations. So let's say this is the donor DNA, represented here on the left by a circular DNA, which is typical for bacterial genome. Well, in its basic form, PCR cloning involves amplifying specific DNA fragments while simultaneously adding linkers, including restriction sites to each end. These restriction sites facilitate the insertion of the amplified DNA into a plasmid vector. During the PCR process, these linkers or restriction sites sequences are added through primers to the ends of the amplified insert, ensuring it can be easily inserted or cloned into the plasmid of interest. So first, the insert DNA is digested with specific restriction enzymes at both sides. This process involves using restriction endonucleases represented here by ND1 upstream and Z1 downstream to cut the DNA at designated sequences, generating compatible ends for ligation. Well, this is just a little view about what you're going to see in details later in this lecture. So the plasmid is also digested with the same pair of restriction enzymes to generate compatible overhanging ends. This compatibility between the insert plasmid ends is crucial for the efficient and accurate ligation of the DNA fragments, ensuring that the insert is properly integrated into the plasmid vector. So once both the insert and plasmid are digested with the same pair of restriction enzymes, we perform ligation to combine the insert of interest to the vector. This process involves using DNA ligase to catalyze the formation of phosphodiester bonds between the 3' hydroxyl and 5' phosphate ends of the DNA fragments. As it's shown here in this scheme, the ligation reaction effectively joins the DNA fragment, creating stable recombinant DNA molecule, which is our recombinant constructs. It is also important to mention that a successful ligation reaction is crucial for creating recombinant DNA. Missing this specific step would compromise the entire experiment. So it's important to ensure it is done correctly. You should make sure also that during the ligation process, the digested insert and vector are mixed together in the presence of a ligase buffer, which is typically contains a fresh ATP to provide the necessary energy for the reaction. As I mentioned previously, cloning primers are quite particular because they need to serve multiple functions simultaneously. On the slide, you can see an overview of the key components involved in designing primers for molecular cloning. First, we have the hybridization sequencing with which are specific regions of the primers that bind to the end of the target sequence of interest. Those sequences play a vital role in the precise amplification of the desired DNA fragment. And next we have the restriction sites, which are sequences recognized by restriction enzymes used to cut DNA at specific location. Adding these restriction sites to the primers facilitate the insertion of the amplified DNA fragment into plasmid vector by creating compatible ends for ligation. It's also crucial to choose the appropriate restriction enzyme to avoid unwanted cuts. and ensure precise insertion. Also, note many restriction enzymes do not cut DNA efficiently at the end of linear sequence. That's why it's advisable to add 3 to 6 bases upstream the restriction site to improve cutting efficiency. These additional bases are known as leader sequence or stuffers. When selecting restriction enzymes, it is important to use first DNA analysis to check that the chosen restriction enzyme do not cut within the insert sequence. And also they do cut at the desired locations in the plasmid vector within the multiple cloning sites. Next, we need to examine DNA sequence that we want to amplify and design primers that we bind to it and replicate it. The figure here shows the end of the operating frame and their use for primer design. We aim to clone an ORF, so we need to clone it from the start codon to the stop codon. A primer of about 18 to 21 base pair is generally sufficient to provide specificity and be compatible with the standard PCR reaction. But we must add ND1 restriction site upstream of the forward primer as well. And of course, without forgetting the stuffer or the leader sequence upstream of the ND1 restriction site. For the reverse primer, the design is quite similar, but we need to use the reverse complement to achieve PCR amplification. We can start similarly by taking the last 18 base of the ORF of interest, including the stop codon, then adding ZO1 recognizing site followed by the stuffer to improve restriction enzyme digestion. So this gives us around 30 base pair sequences with 18 base pair of homology with the ORF. When we have these sequences, we then need to generate the reverse complement of the reverse primer to successfully amplify the ORF. We can generate the reverse complement using one of the free online tools. If we input the sequence chosen for our reverse primer into one of these calculators, we obtain a final reverse primer sequence. Now that our specific cloning primers are ready, the only thing left to do is to synthesize or order them and start cloning. Once the primers are designed and synthesized, we proceed to PCR amplification to obtain the full sequence, including linkers at both sides and also to produce sufficient number of DNA copies for easy handling. It's crucial here to use a high fidelity polymerase to avoid mutations. The longer the expected PCR product, the more important the fidelity of polymerase becomes. Also, choose a primer hybridization temperature close to its melting temperature for higher specificity. After PCR amplification, the PCR products are purified using a commercial kit and then performing quality control as well as sequencing to check that our insert sequence is here, which is something very important to do at this stage to ensure you have your gene of interest cloned and expressed. Once the PCR amplification is complete and PCR products are purified, the next step is the digestion of both PCR product and the recipient plasmid using same pre-selected restriction enzymes. This step is crucial because it ensures that the end generated are compatible for the sequence ligation process. The insert is then ligated in the plasmid. For a standard ligation reaction, we usually use around 100 nanogram of total DNA, and it's recommended to aim for 1:3 molar ratio of vector to insert, because at this ratio, it's optimal for efficient ligation. And then remember, the ligase buffer should be fresh to maintain ATP integrity, which is crucial for the ligation reaction. Once the ligation reaction is complete, the next step is transformation, where we introduce the recombinant DNA into competent cell. For most standard cloning procedure, you can transfer 1 to 2 microliters of the ligation reaction into competent cells such as DH5 alpha as a library or BL21 as an expression strain. In the transformation process, competent cells are prepared by treating them with calcium chloride containing calcium ions, which is very important. The calcium ions plays a crucial role in this process. They help to neutralize the negative charges on the DNA backbone and the cell membrane, allowing the plasmid DNA to come closer to the cell surface. The illustration shows a close-up of competent E. coli cells suspended in calcium chloride solution. The circular structure within the solution represents plasmid DNA, which is our recombinant construct. The calcium ions present in the solution play a crucial role in the transformation process by facilitating the binding of the plasmid to the bacterial cell walls. During the transformation, the cells are typically subjected to a heat shock, where they are briefly exposed to higher temperature. This heat shock creates a thermal imbalance that temporarily opens pores in the bacterial cell membrane, allowing the plasmid to enter the cells. After the heat shock, the cells are returned to a cold environment on ice, which helps close the pores in the cells and trapping the plasmid DNA inside. After the DNA is being introduced into the cell or competent cells, enrichment medium is added and the cells are incubated with shaking. Actually, this incubation is crucial for 2 main reasons. During this time, the cells recover from transformation process, repairing their cell walls and membrane. It enables the expression of antibiotic-resistant gene. This expression is mandatory for selecting successfully transformed cells, as only those with the plasmid will survive on antibiotic-containing media. The plasmid DNA can replicate within the bacterial cells, leading to the expression of the antibiotic-resistant gene carried by plasmid. Regarding plasmid replication, it's important to note that this replication can either be concurrent or not with bacterial growth. In fact, the rate of plasmid replication can vary depending on some factors, including the type of replication, origin, and the growth conditions. For instance, plasmid with a high copy number origin of replication can replicate more quickly, resulting in a higher number of plasmid copies per cell. And the plasmid with low copy number origin replicates more slowly, producing fewer copies per cell. Also, growth conditions such as nutrient availability, temperature, and other environmental factors can influence the rate of plasmid replication. After the incubation period, the cells are plated on LB-containing antibiotics. Only bacteria that have successfully taken up the plasmid DNA which includes the antibiotic resistant gene, will be able to grow on and form colonies. The selective pressure ensures that only the transformed cells are able to proliferate, allowing for easy identification and isolation of positive clones harboring gene of interest. The number of bacterial colonies resulting from the transformation will give us an estimation of the transformation efficiency. Ideally, the recombinant plasmid should result in significantly higher number of colonies compared to the non-recombinant plasmid. This difference indicates successful insertion of the DNA fragment into the plasmid. But having explored the PCR-based cloning workflow, let's now move on to the next section, which is the heterologous expression of recombinant proteins. Heterologous expression refers to the introduction of a gene from one organism into a different host organism to produce specific protein of interest. This technique allows researchers and allows us to study proteins in controlled environments and to produce large quantities of proteins, even those that may be difficult to extract from their native sources. Let's look at the different systems we use for protein expression, which falls into main category, cellular system and transgenic organism. Cellular systems are the most common platform and seem to be the best and easiest way to express recombinant protein. Sorry, I am here again. So I try to start over from
insect mammalian cells.
So I'm here again. Can you hear me?
No, we can hear you.
Yeah, I have missed a lot of things.
Well, go back to the slide when you have bacterial, yeast, insect, and mammalian cells.
Okay, this is, yeah, okay. Okay. Well, you can see the presentation?
Yes, we do.
Thank you. So let's go back to the our presentation. So I have been talking about cellular system. I think we have lost the connection here. So I have been saying that cellular systems are the most common. I think I've been stopping at the use of yeast cells. So I have said that yeast cells can provide accurate protein modifications. And I'm saying that Saccharomyces systems are relatively easy to handle and offer a middle ground between the bacteria and the complex system. So in certain cases, I said the use of insect cells seems to be useful as well. We have the baculovirus, for instance, which is ideal for producing complex eukaryotic proteins capable of extensive post-translational modifications. It is especially useful for proteins that are toxic to other hosts. On the other hand, and as the top choice for therapeutic proteins intended for human use, mammalian cells like the Chinese hamster ovary are the most used. They can perform human-like modifications, ensuring the correct structure and function of protein. We have also the second main category, but it will be more detailed today. It's the transgenic organism system. This system can offer scalable solutions, but they may involve many ethical and regulatory considerations. For the remainder of the lecture, we focus solely on bacterial system as we do not have sufficient time to develop all systems in details. While vectors come in many forms, when we focus on bacterial systems, one class of vectors stand out for its widespread utility and effectiveness. They are plasmid vectors. Among the various plasmid vectors, expression plasmids are particularly significant. These specialized vectors are engineered to ensure the efficient production of recombinant proteins. Key elements of these vectors include the origin of replication, repressor, selectable marker genes, and also multiple cloning site here. The multiple cloning site is flanked with regulatory elements, including promoter and terminator sequences. Also, the vector should contain ribosome binding site for efficient ribosome recognition, and also usually contain fusion tags such as His tag, which plays a significant role in the purification and solubility of recombinant proteins. However, However, the fusion tag must be carefully selected to avoid disrupting protein function. Another important factor is codon preferences. Adjusting the codon usage to match the host's tRNA availability can significantly enhance protein expression. This adjustment can be achieved by modifying the gene codon sequence or using genetically engineered Ecology strain that efficiently translates RAR codon. Ecology is one of the most popular hosts for the overexpression of recombinant proteins due to its rapid and efficient growth. The most widely used Ecology strains in heterologous protein expression are BL21 and its derivatives. This is primarily because BL21 strains are deficient in cytoplasmic proteases and outer membrane proteases as well. The Ecology BL21 DH3 strain is cornerstone protein expression technology. This strain is particularly unique because it carries the T7 RNA polymerase gene due to the lambda D3 bacteriophage. Essentially, this gene is under the control of the lac operon, meaning its expression can be induced by the presence of lactose analog, which is IPTG. Now, when we induce this system, the T7 RNA polymerase is produced, which in turn drives the high-level expression of gene placed under the T7 promoter. This makes the BL21D3 strain efficient for producing recombinant proteins. Also, there are two primary variants of this strain to consider. The BL21D3, which is our go-to for general protein expression test because it's highly efficient and widely used. And we have the BL21D3 P lysis, which contain an additional plasmid P lysis. Let's talk first about the BL21D3 strand. This, I will use the pointer. Well, this strain has the gene encoding the T7 RNA polymerase integrated in its genome. So when we introduce a T7 expression vector in this strain, such as PET vectors, the T7 RNA polymerase, which is produced by the host cell, recognizes the T7 promoter on the vector. This interaction initiates the transcription of the gene of interest, leading to the production of the target protein. The BL21D3 P lysis, on the other hand, is quite similar to BL21, but with an additional plasmid P lysis. The P lysis plasmid produces the T7 lysosome, which inhibits the T7 RNA polymerase. Well, this inhibition is crucial because it helps control overexpression of target protein, preventing potential toxicity or metabolic burden on the host cell. This strain is commonly used to express toxic or large proteins without inhibiting the growth of the host cell. Even though it's considered as an excellent expression system, Ecology still has some limitations. The cytoplasmic environment of Ecology is a reducing environment, which is not conducive to the formation of disulfide bonds. Why those bonds are crucial for stability and functionality of many proteins. While the reducing environment can facilitate the production of soluble proteins, it often complicates the purification process as well. The major issue here is the formation of inclusion bodies, which are dense and soluble aggregates of misfolded proteins. Although they are easily isolable and protected from proteases, their solubility necessitates refolding to regain functionality. At the periplasmic space level here, proteins typically have a signal peptide directing them there. Contrary to the cytoplasm, the periplasmic space is an oxidizing environment which appears to be favorable for the sulfide bound formation. Also, this compartment contains fewer proteins than the cytoplasm, which simplifies purification. It is also important to mention that in E. coli culture, recombinant proteins are not secreted in the culture medium, which implies the need for cell lysis to release the expressed protein. While E. coli can achieve high expression yield, one major issue I already mentioned is that large proteins often precipitate and form inclusion bodies. So to address this, two key strategies have been developed. The first is dissolution and refolding, where inclusion bodies are dissolved in strong denaturant like 8 molar urea or 6 molar guanidine hydroxide, which disrupt the non-covalent bonds holding the protein aggregates together. The second strategy here involves modifying the culture conditions to prevent the formation of inclusion bodies. This is actually achieved by reducing the production culture temperature. For instance, in E. coli, protein expression is often conducted at 28 degrees Celsius instead of 37 degrees Celsius. Lowering this temperature slows down protein synthesis, giving the nascent polypeptide chains more time to fold properly and enhance the folding environment, which reduces the likelihood of misfolding and aggregation. Let's wrap our lecture by diving into the key steps of purifying recombinant proteins, starting from expression testers and until affinity purification, all in one single last slide. Well, if we want to purify our recombinant protein, we need first to check proper expression. Therefore, we start usually with expression testers where small-scale cultures of E. coli are grown and protein expression is induced under various conditions to identify the optimal settings for maximum protein yield and solubility. To verify protein expression, we use SDS page, so we have an idea about the size, which provide us with a clear indication of whether the recombinant protein is being processed and expressed or not. Once optimal conditions are identified, we move to scalable production. This step involves cultivating the host cells in bioreactor under controlled conditions. Bioreactor here provide an ideal environment for microbial growth and protein production by maintaining precise control over parameters such as pH, temperature, and oxygen levels. After sufficient growth and protein expression, the cells are harvested and lysed, and you proceed to the affinity purification. This method leverages the specific binding properties of affinity tags, such as the His tag, which is commonly used due to its strong affinity for metal ions like nickel ions. The harvested cells are lysed to release the intracellular proteins. And the lysate is passed through the colon containing this resin with immobilized metal ions. So the His target protein binds to the resin while other proteins are washed away. And finally, the target protein is eluted to releasing the purified protein. And that's how we obtain our final product by cloning and heterologous expression. And that's it for today. Thank you for your attention. شكرا على حسن الإصغاء. I hope the concepts we explored today have provided you with valuable insights and a little bit deeper understanding of the cloning. And I encourage you to reflect on today's lecture and apply these ideas in your research and practice. And I look forward to our next lecture. And until then, please feel free to reach out with questions and thoughts. And thank you. I think you are hearing me, Slavi?
Thank you very much for the presentation and very detailed explanation what's going, how it's working, what's the steps and how we can do this process. So do you have any questions? Any questions from the chat system? What do you think? I actually have a question. Since you work with the bacteriocins, what do you think, what kind of host will be the best one to express the bacteriocin genes?
Slavia, I have already done that. The best host was the BL21D3 P lysis because the bacteriocins have impact on the E. coli. So the overexpression with the BL21D3 was a little bit impacting the growth of this strain. So I have used it for the next expression, the BL21P lysis. which is the best in this case.
And do you face some problem of the stability to host to keep the gene to express it? Because when we was cloning one of our bacteriocins in the Saccharomyces cerevisiae, we was actually facing this problem because after a couple of weeks we was losing the expression. So we needed to actually constantly keep the supplying the Saccharomyces with the cloned gene.
Actually, I have used the libraries, and I have already said it was the BL21DH alpha. This strain is actually very stable. So when I cloned for the first time my gene, I put it there and I conserve this. I stock it at minus 80, and wherever I want to express it, I take it out from the cold storage and transform it in BL21D3 pelases again and express my protein with all ease of the as it is.
I think the experience is very important because as I very often say, no general recipes, everything is art, and we need to combine the several approaches to achieve our goal. So, but experience is one of the more important because in this way we can avoid some of the common mistakes. So thank you very much for the presentation.
Welcome, thank you to you, Mr. Todorov, and see you the next time.
Maybe in Algiers in October, maybe.
Yes, yeah, for sure. And maybe I will be there to see our PhD student in presence. That will be better if we do some workshop in there.
Okay. Thank you.
Thank you. See you. Thank you. Bye.
And I will share your email with our audience. So if they have any questions, they can email you directly.
Anytime. Okay. You will be very welcome.
Thank you very much for the presentation.
Thank you.
And we're going to have a lunch break now. So please, we can be back at 1 o'clock, 13 p.m. to continue with the presentation of Kristina Boksan. And then we have Nadia Boyko from Ukraine. Okay, thank you very much and see you in 1 hour. Hello, everyone. Can you hear me? Please, if you can hear well, show in the chat system that everything's okay with the audio. So the next presenter. Hello, Chris.
Hi, how are you? Yes, I'm hearing
we
you.
are all
Are
of
you
us
hearing
are
me?
very good because we're in Brazil and we enjoy the Brazilian winter. And you are in the very warm US, so you're boiling
Yes.
there. It's a very big pleasure to present the next presenter in these seminars. It's Professor Cristina Boxon. She will talk about microRNA. She is currently at University of Harvard, right? Are you there?
Yes, I'm still here.
So she's a professor at Faculty of Pharmaceutical Sciences of the University of São Paulo since 2050, and she worked in the food technology department. She graduated in pharmacy and biochemistry from the Faculty of Pharmacy and Biochemistry at University Paulista in 1999, last century. He held a master's degree in microbiology and immunology from the Federal University of São Paulo, 2002, and then a PhD in science from the University of São Paulo in 2012. Her career out teaching and research activity as a PhD professor at the area of food technology at the Department of Biochemical Technology, Pharmaceutical Sciences, Faculty of Pharmaceutical Sciences, University of São Paulo, since 2015. And currently, she is a visiting professor in Harvard Medical School at the Institute of RNA Medicine. He has experience in area of microbiology and immunology, extracellular vesicles, and the food science and technology, working on the terms functional characterization of traditional fermented foods and development of functional characterization of and study of the mechanism of action in new probiotic products. It's a very big pleasure to have you today with us, and we are looking for your presentation. And if I know that your personal preference for Portuguese or French, you still need to speak in English because we have a lot of participants from all around the world. So thank you very much to be with us, and please, you can start with your presentation.
Thank you for having me. It's my pleasure to be here and share a little bit of knowledge. I will talk about fermented food and microRNAs. My field is probiotics and microRNAs now, that's what I'm studying here. As Professor Slav told, I'm professor from Faculdade de Ciências Farmacêuticas, but I'm working here in Harvard Med School. I'm doing my postdoc here. So it was my pleasure to share with you a little bit of my knowledge. So I will show a little bit what is fermented food, the safety aspects of and beneficial properties of fermented foods that I believe was largely discussed before. And then I will introduce what are microRNAs, how the microRNAs act in food and nutrition, and how are the future directions in this field. Fermented foods are made through desired microbial growth and enzymatical conversion of food components. So the fermented food is not that food that fermented for any microorganism. It should be a specific microorganism that you have interest to grow and receive the metabolites produced for that microorganism. The fermentation also have been used to preserve and transform foods many years ago. Since Babylonia, we notice the fermented food in the diet of the people. For this reason, the fermentation creates more stable and diverse foods with unique organoleptic texture and functional properties. Have some key microorganisms that we use to work in food, and they could be bacteria like Lactobacillus acidophilus, Lactococcus Thermophilus, like Bifidobacterium animalis, could be mold, could be yeast, could be some fungi, could be specific microorganisms that we already know, and some that we still discovering, but should be well characterized. The common types of fermentation, we have many types, and the types of fermentation are based on the end product format. So we have the lactic acid fermentation that produce yogurt from lactic acid bacteria. We have alcoholic fermentation from some yeasts like Saccharomyces that produce CO2 and alcohol. You have acidic fermentation produced by Acetobacter bacteria that will produce acid acidic. So we have some kind of fermentation that is classified for the end product format. Some safety aspects of fermented food is the preservation method. So you can prolong the shelf life of this product. As example, the milk. The milk have a shelf life of 2 or 3 days under refrigeration, but after fermentation, you can prolong to 40 days under refrigeration. So you keep a very long time of preservation of this food. You reduce the risks of contamination and risk of development of pathogenic bacteria, because when you ferment, you decrease the pH of that food, and when you reach some pH above 4.5, then the pathogenic bacteria have more difficult to grow, so you decrease the counting of pathogenic bacteria, reducing the risk of this food produce pathogenic things for our health. Also, they improve food flavor and quality. So we have some flavors that are acquired just through fermentation. Some umami flavors, some acidic flavors that could be acquired through these microorganisms that use the food as substrate. The main beneficial properties of fermented food, they are promote bowel regularity and digestion. They support a healthy mood, because some microorganisms produce some neurotransmitters that could improve the mood. They help in cognitive function also for the neurotransmitters. You can bring strong bones for increase the calcium absorption. You can promote cardiac metabolic health through some fatty acids that they are formed during the fermentation. They regulate cell growth. They boost the immunity and decrease inflammation, and they can support our gut microbiome and other benefits that the fermented foods could bring. So they improve bioavailability also of nutrients, synthesis of vitamins and minerals, and promote probiotic effect on gut health. Fermented food are not probiotic food. They could be probiotic food, but not all fermented food are probiotic food. So some microorganisms that are in the that you use during the fermentation could be probiotics, and then you have a probiotic fermented food. But not all of them are probiotics. And they promote immunomodulatory effects and have potential anti-cancer properties. To produce all these beneficial effects, some of them like immunological effect, anti-cancer properties, modulation of the microbiome. Some of them could be modulated through epigenetic modification. What is epigenetic modification? This is when you change the expression of your genes without change your DNA. You have your DNA, then you translate to your RNA, and then you traduce to your protein. Before you traduce to your protein, because the protein that will be the modulation of the pathway that we are doing, you can interfere in this. And then the interference between the RNA and the protein we call epigenetic modification. This could be through DNA methylation that implies in not translate the gene, a stone methylation, a stone acetylation, and through no coding RNAs that block the gene expression after the RNA be traduced. Then, for the non-coding RNA, we have many types of no-coding RNAs. These no-coding RNAs are functional RNA molecules that don't translate into proteins. They play vital roles in cellular process, including transcription and translation. And some examples that we have is the transfer RNAs, small nuclear RNAs, ribosome RNAs, long no-coding RNAs, or small no-coding RNAs, but the classification of small or long is the number of base pairs that they have. Today we will talk for the small non-coding RNAs, and specifically from the microRNAs. What is why we are talking about microRNAs when we are talking about food? Because microRNAs could be a bioactive component, and could be a bioactive component that we find in food. So they are involved in the regulation of a variety of biological processes. They interfere in differentiation of T cell, B cell, dendritic cells, and macrophages. They interfere in proliferation and cell death and differentiation of the cells. They improve or not the release of inflammatory mediators. So the microRNAs could interfere in all these health benefits that we see in our food. But what are these microRNAs? So microRNAs is a single-strand no-coding RNA molecule that is approximately 22 nucleotides. They have a variation to 18 until 24 nucleotides in length, and they are very conserved during all species. So we have a similarity in these microRNAs, these no-coding RNAs, since the mold, the fungal, until the human being. And we have some microRNAs that is conserved in all species. We don't have microRNAs on prokaryotes, but we have some no-coding RNAs that are very similar to microRNAs. To be microRNA, they are produced in the nucleus, a pre-microRNA. Then the drasha transform in pre-microRNA. They are transported out of the nucleus to the cytoplasm through the transportin 5. Then the dicer complex with this pre-microRNA and cut in double-strand microRNA, and then the strands are cleaved, and one strand complex with RISC and Argonaut proteins, and complex with the messenger RNA blocking the translation of this microRNA. So when they have the paired, but not fully paired with the target messenger RNA, we just repress the translation. Then you block here, and then you don't translate the protein. But when you have full connection, a full connection of the microRNA and the target messenger RNA, then we have the degradation of this messenger RNA. So the microRNA could act in many messenger RNAs, and some they just degrade the messenger, and then you will not transcript anymore, and some you will just repress. So if you take out this complex risk microRNA, this target messenger RNA could be translated again. And you can, one microRNA could target many messenger RNAs. This is a new field. They started in 1993 when first was discovered the microRNA Lin4 in C. elegans. And then they discovered that these microRNAs could be extracellular and could be in plants, not just in worms or in flies, but also in plants. Then in 1998, the first observation of RNA challenging the central dogma of biology that brings the Nobel Prize to Farrow and Mello in 2006. And then we have the first human microRNA identified, that was the LAT7 in 2000. And then we start to discover the interference of this microRNA, that they are involved in cancer, they are involved in aging, they are involved in inflammatory regulation, they are involved in apoptosis of some cells, and then they start to see the signature of some microRNAs to diagnose a specific disease, and how to bring health when we diagnose one microRNA as a signature of this disease. Then they start to work to provide these microRNAs for the people that suffer from this disease, and then they start to work with the personalized medicine. And then they start to identify how these microRNAs could do the cell-cell talk, and how they can reach different tissues and organs, and not just cell-cell contact talk. And then the field will grow exponentially, and today we have the personalized medicine that we can deliver specific microRNAs for the specific disease and alleviate the symptoms of this disease, and promote a regression of some disease, specifically in cancer field. And what is this is important for food? Why we are studying microRNAs if all the research are on disease treatments and focus on cancer? Actually, we know that everything can express microRNAs. All food can express microRNAs, and we eat this every day. So we eat a lot of microRNAs from our diet that could interfere in our genes. So how we can improve, how can we improve our diet benefits? Through the microRNAs that we already know that we are intaking. So we know that these microRNAs from the food, that some authors call exo microRNAs, some people call diet microRNAs, they can complex with vitamins and minerals, and they can complex with the risk of the cells. They can be internalized in our cells and complex in the risk and argonaut complex and interfere in the target messenger RNA of the host. Also, we can modulate how the microRNA will be expressed in our cells. So we know that some eggs, for example, have high expression of MIR142 and 1A1B and MIR30A5P. We know that meat have MIR1, MIR99A, apple have MIR7121DH, broccolis have MIR159. And when we intake all of this food and we are intaking all of these RNA, what happens in our body? Part of them go to bolus, fecal bolus, and then they are excreted. They are not absorbed, and part of them are absorbed. And when they are absorbed through the intestinal epithelial cells, they go to the blood vessels, and then they can reach several organs and distant tissues and do the effect that we want or not, depending on the microRNA that you are looking for. And then they can exert epigenetic interference in some diseases, in some disorders. For example, the MIR30A, they increase the osteoclast formation. Then when you intake eggs and you intake MIR30A, and they are absorbed, then you can improve your bone health. It's one of the examples of what happened. Also, when they are here in the bolus, when they are here in the bolus, in the fecal content, they interfere in the microbiome. So these microRNAs that came from the diet, they are here in the fecal area, and then they can interact with the bacteria. When they interact with the bacteria, they are absorbed by the bacteria, and they can not just get the RNA, but they can get the DNA, because bacteria don't have nucleus. So you can interfere the translation and the traduction of this bacteria in a way that you can modulate your microbiome. You can grow better or not this bacteria, and you can change the metabolites that we are producing. Remember, in the fermentation, we are looking for the metabolites produced for that bacteria, specific bacteria. When you can modulate the bacteria through the microRNA that is in the same content of this bacteria in the microbiome, we are modulating our microbiome, not just from our food, but also for a specific microRNA. And this is important when we think in future direction, when we think in biotechnology, that you can address during the fermentation the production of the microRNA of your interest to modulate the microbiome of someone that have a inflammatory bowel disease, for example, and alleviate the symptoms, regulating this microbiome. Also, the bacteria produce some non-coding RNA that could interfere with epithelial cells. So it's not just the epithelial cells that produce the microRNAs that came through the luminal and interact with bacteria, and the diet microRNA that interact with the bacteria, but also the bacteria produce some non-coding RNAs that could do the other interaction. So to understand what happened, when we have the mice, one mice that is conventional and one mice that is germ-free, don't have any gut bacteria, and you inoculate Listeria monocytogenes. The conventional mice have an expression down-regulated of MIR143, 148, 200B, 200C. And when you go to the germ-free that don't have other bacteria, don't have the microbiome established, and have just the Listeria monocytogenes, the expression of the microRNAs that we collect in the feces are totally different. The microRNA 143, 148, 200B, and 200C don't have any change when you get the feces before and after the inoculation of Listeria. And for the MIR 194, they have a down-regulated, but to the 378, they have an up-regulate. So the microbiome and the bacteria that you introduce in your gut, they will change the expression of microRNA that you can see. This expression actually is not in the fecal content. This expression, they got the serum of these mice and analyzed the microRNA content in the serum of these mice after the infection. Also, when you work with probiotics and microRNA, you can see that not all probiotics have all the same effects. We have a probiotic, a strain-dependent manner and a time-dependent manner. So when we work with 3 different probiotics in different times to analyze the expression of MIR 148A in CACO2 intestinal cells in vitro, you can see the expression of this microRNA changes during the time and and the kind of probiotic you use it. So the modulation of our microRNAs depends not just of the diet, but depends of the microbiome and you can change what they are expressing as we know what we are eating and we know what we want to modulate. Also, the infection, when you infect the mice and you give probiotics, different strains of probiotics, and then you infect these mice, you can see the modulation of these microRNAs are totally different through the time of post-infection. So you stabilize the probiotic, you know how you will modulate your microRNA and which outcome you will have. Then you infect the mice, they still get a different modulation through the time. So when we analyze to produce a new product, a new functional product, looking for the modulation, for the epigenetic interference, we have to analyze everything, all the food matrix, what have in that food matrix, what have in the microorganisms that we use to formulate this fermentation, and what will happen in our system. So we know that these microRNAs from the diet, they can modulate the epithelial cells, they can modulate the microbiome. But how they can modulate? Here, when they are just expressed like this, they could be complex in some proteins, but when they are complex in proteins, they are degraded for all enzymes that we have. When we start to eat, and during the oral phase that you have many enzymes, they start to degrade these microRNAs. They are stable, but not that stable that can support the PGA passage. So they discovered that to be absorbed, mainly they have to be encapsulated, and our body do this encapsulation. So to have to be absorbed, and then by activity, how they do? We know that the microbes can interfere in this expression of microRNAs. The epithelial cells can secrete these microRNAs, and normally they are secreted encapsulated in extracellular vesicles and then modulate the microbiomes. The diet can interfere in these microbes, that the metabolites of these microbes can interfere in the synthesis of the expression of these microRNAs, and also the dietary microRNAs from the extracellular vesicles can interfere from microbes that interfere in epithelial cells. And this is important because when you are doing a new product and you want to target this product to a specific microRNA, we can encapsulate this microRNA and then ferment it and produce our food with these specific microRNAs encapsulated in the food, and then they deliver for our for our patient the food with a high expression of that microRNA of interest. So what are the extracellular vesicles? The extracellular vesicles could be some kilomicrons, some particles of fat particles, could be apoptotic cells. All of them are vesicles, but extracellular vesicles that we have interest, they are apoptotic bodies, microvesicles, and exosomes. And these, the classification of them is depend of the size of the particles and the way that these particles are produced and deliver the microRNA that is encapsulated here. And when they are in these extracellular vesicles, they can just pass through the TGI. So during the ingestion, they can pass, they survive to acidic and enzymatic effects, and then they can reach the bloodstream and migrate for all other tissues in our body. So for example, the milk, that was what I studied, the milk, all microRNA that the content of the milk, they are destroyed during the TGI, except for the microRNA that are encapsulated in the exosomes. Everything what is in the exosomes could pass through the TGI and be absorbed and go to blood circulation. And then the microRNA modulation, they can improve the phagosomal maturation through the MIR21, 3P, and 30B, 5P. They can produce more nitric oxide. They can promote the apoptosis through LET7E. They can promote the autophagy, improving the MIR33 expression. They can modulate the immune response through signaling to improve the cytokines release through antimicrobial peptides, improving the antigen presentation. So each microRNA have a specific pathway, but if you can see here, MIR21, they can improve the phagosomal maturation, the apoptosis, the antimicrobial peptides, and then, we know that one microRNA have many targets. So when we draw a new functional food based on the microRNA that we want to improve the expression, and you want to put in your food, you have to analyze all of targets that microRNA could reach. And this is the high difficult to precision medicine and to precision nutrition, because until 4 years ago, we were talking about precision nutrition when we start to study microRNA. precision medicine, but now we start to talk about precision nutrition, when we can modulate specific disease and decrease the symptoms, and sometimes do the therapeutics of this disease through specific food, that we can modulate this. And then what are the future directions? We have a potential for new functional foods based on this, and the implication for personalized nutrition and medicine. So this is the main topic that I bring for you. And now I open to question, and I open to the floor to the questions.
Presentation. You cannot see me on the screen because I'm hiding here, because I want to read the question from the chat system. I think I can start with the question of Wellington here. He was asking, how stable are these microRNAs? I was told that RNA molecules were quite unstable molecules. Seeing them in the gut and affecting the gene expression in bacteria, it's quite astonishing. So what about their stability?
Now they are not that stable. MicroRNAs, they are a little bit more stable than the RNAs, that because they are very, very small. But in the TGI, they are not stable. All the enzymes that we have a lot of RNases that degrade the microRNAs. This is the reason that when you secrete and you are cell close to cell, then they can exert some effect, but not from the tissue to exert the effect that we are looking for when we talk about diet RNAs. This is the reason this field was so controversial for many years, because they say, okay, we found the microRNAs on the food, but they are degraded very easy for the RNases, but they are encapsulated for these extracellular vesicles, and the extracellular vesicles are really stable. You can do all the digestion and they are stable, and they can reach the cell, reach the tissue, they can be absorbed and go to the bloodstream and then do the effect. These exosomes, they have a Catherine system that they are absorbed for the cell, the host cell, and they are engulfed for the bacteria. Many extracellular vesicles are engulfed for the bacteria and internalized, and then they set the effect inside the bacteria. They change completely the metabolites they are producing. They change the time of fermentation. When you include some extracellular vesicles that you transfect with the specific microRNAs and put in your food matrix and put your bacteria to ferment, they ferment differently.
We have another question from Icaro. I think we lost your audio. Can you hear me?
You lost my audio? Yes, I'm hearing you.
Okay. It's a question from Icaro Oliveira. How do environmental factors such as temperature, pH, and nutrient variability affect the expression of microRNA-like molecules in the microorganism involved in the fermentation?
They affect temperature, pH, and nutrients availability will affect the bacteria or the microorganism during the fermentation, and this will change the expression of the microRNA. When you have a microRNA that is encapsulated in an extracellular vesicle and added to your environment, then we know that some particles, they have a different stability during the pH variation. So we know that they tend to aggregate and then get stability again. And sometimes these particles can just change the content of the particle during this pH variation. And this is one fact that we have to study when you are getting another product, when you are drawing your product, because if you do the fermentation process, you have to analyze each point of your fermentation curve to know if the vesicle that you input in the beginning is still the same vesicle that you put in the end. Then how much that microRNA will be delivered for your patient. They can affect.
And
I don't know if I answer you.
Yeah, well, it's not me, Ikaru, he was asking. And I have a question for you. What about the microRNA from the spoilage and foodborne pathogens, or even another pathogens, how they can actually interfere in all this process?
They interfere a lot. Many times they are modulating our cells through this interference. As the good bacteria interfere, the bad bacteria interfere the same way. This is the reason that I show the here. Let's see. When we analyze here the Listeria, but we just put the Listeria, and Listeria change all expression of the microRNA in the serum of these mice, and this is correlated to microbiome. When you are the normal microbiome, it's your body reacts in a way when you don't have this microbiome well established, and then you get the pathogenic bacteria. They express in a different way.
And now I will ask Wellington to read another question from the chat system, because I'm sitting in the front, he's in the front of the computer. So Wellington, can you please... No, you cannot see him, but you can hear his voice.
No, I'm right here.
Hi, Wellington, how are you?
Hi, good, and you? Thank you for your talk.
Good.
Very interesting. I'm just curious.
Thank you for having me.
So as I see, it's a two-way communication, sort of interference, I guess, because you have microRNAs from host cells going to the bacterium, but you can also have small RNAs from the bacterium going to the host, right?
Yes, right.
Okay. And from the host to the bacterium, I was just curious. So these microRNAs are like transported via vesicles, which are probably membrane-like vesicles. So is there a difference between the action of these vesicles in gram-positives and gram-negatives? I mean, because the cell walls are different.
They are totally different. We have, from the bacteria, we don't call, they are extracellular vesicles, but when we talk about extracellular vesicles from the food, mainly they are exosomes, they are very small. But from the bacteria, we call just extracellular vesicles, that is membrane extracellular vesicles. For gram positive, we have just one membrane, then they get the cytoplasm content, and then they deliver for the host. When you have the gram-negative bacteria, you have double membrane. Then you get a vesicle that have double membrane. Then you have the cytoplasmic microRNA inside of the inner membrane, and then you have the out membrane, between the out membrane and inner membrane, and this gram-negative membrane vesicle that also have some microRNAs that could be there. And then the modulation is different. This is a field that they still looking for, have many doubts. they are not totally sure about what happened with them, but they know that these membrane vesicles modulate the epithelial cell and can silence the immune response, allowing the pathogenic bacteria to spread.
Or maybe they're using some of the already pre-set channels in the cell wall and membrane.
Mm-hmm.
Well, because we know that some of the, actually, when you have transformation of some DNA from one to another, sometimes they can using already existing channels in the bacteria. Maybe.
Yes, but they are not, okay, these vesicles also transport DNA, transport other kind of no-coding RNAs. They transport RNA, transfer RNAs, they transport ribosomal RNA, they transport part of proteins, peptides. Then sometimes it's not just the microRNA that is inside of these vesicles from the bacteria and from the cells, have many other things in the content.
They can maybe incorporate the phage RNA or not? Can microRNA can be incorporated in the phage RNA and then not? I'm just.
I don't know. I never saw that. I never read anything about phages, but I didn't look for. But okay. I can look for. I don't know.
There's more questions from the audience. Be careful. She can be in your defense soon. So rather ask now questions. Okay. Thank you very much, Chris, for the presentation. Enjoy the warm weather. We are struggling with the cold weather here. But hope to see you soon. Have a nice time, and thank you very much again for your presentation.
Thank you for having me. I appreciate very much the invitation. Thank you.
Okay, we're going to have some commercial break, and then we continue at 2 o'clock with the presentation of Professor Nadia Boyko from Ukraine. And so it's time to grab the coffee and because then it's a marathon of the Ukrainian presentation at 2 o'clock.
Hello, everybody.
Hello, Nadia. Nice to see you. Can you hear me well there?
Me too. So,
looking? Can you hear me?
yes, I'm hear you very well. And I hope you see already my presentation, I guess.
Okay, but first let me tell the people about you, because it's a pleasure to have next presenter, Nadia Boyko. She's from Ukraine, and she's a head of the department, professor at University of Uzhhorod National University. It's located not too far away with the border with Slovenia, right?
Slovakia.
No, Slovakia. Sorry, always a problem, Slovakia, Slovenia. And she's a principal investigator. She's involved in several European projects, been a visiting scholar in the US, several European countries. Actually, reading your entire CV will take all the time of your presentation, because it's really a lot of activities. If you want something to be done right, work with her and will be done. And she's going to talk a very delicate subject, actually using of probiotics and how we can control actually stress, because it's a more and more important topic for everyone around the world. So thank you to be with us today. You can now share your presentation, and it's time for you to talk about your experience. Thank you very much.
Thank you so much. So, yes, in my presentation, as you said, first of all, I would like to thank you for invitation. And yes, I hope you can see very well presentation. And actually, it has at least a few titles. And in majority, yes, I would like to at least to answer the question whether probiotics we really can use for something good, let's say for control of stress. But from another point of view, as you mentioned...
Nadia,
yes?
Sorry, can you put it as a full screen? Because now it's appearing and don't moving the slides. Just see the title. Just do it as a full screen.
Is it better?
First made it a full screen and then share.
Okay. Can you see it now, no?
Not yet.
Okay, just a minute, please.
Now we see only you.
Can you see it now?
Not yet. No, but again, it's not a full screen. It's just the first slides. And we had a small slides in the site.
Okay.
So it's another stress.
Yes, exactly. You're right. Because, yeah, actually, it's on my screen. It's completely on an old screen. But when I putting share screen, it's asking me.
The specific one that is a full one. When you select from the options.
Yes, I will. Let's see what happened. Is better now?
No, it's nothing. It's just your video.
Okay.
Sorry.
No, it's okay. And now?
Try to change slides, please. Yeah, it's changing. Okay, now it's great. Perfect. It's working. Thank you.
Okay, thank you so much. So again, we would like, I actually would like to speak with you about probiotics, whether we can use them for control of stress. Otherwise, I also will be trying to touch a quite sensitive subject. Can we really come with the new ideas, if we learn very deeply to improve somehow the mental health and also recovering and rehabilitation of Ukrainians, since, let's say, Ukrainians now is a very specific situation, as you probably know. So can you see my next slide? Is it moving? Can you see analytic? So when you see analytic today, then
yes,
is it working? Yes?
Yes, it's working. We're on the second slide.
Okay, thank you. Sorry, I need to ask before I will go ahead. So actually, we were a quite big country. We have 36 million people. But now when we come to estimation based on Institute of Demography and Quality of Life problems, we already learned that if we will go this way, probably we will have a huge decrease in the next 26 years. So we will come to only 25 million. And actually, why it happened, because it's already, we lost already approximately, if you can see from this number, we already lost around 5 million of people. So actually, this is different issues, because first of all, we have a huge movement of population. There are a lot of people in mainly women and children are currently in forced emigration, what is actually connected to the war situation. But is also another issue which I would like to point it out, that we have a very huge problem with the average life expectancy, because now average life for men, before it was 66 years in 2020, but now it's come to 57. And the same is for women, actually, so it's not connected exactly for a soldier. I mean, it's women which lived, they have an average of living 76 years, but now they have only 70. So actually, the situation we have, as I mentioned, it's not very good, because it's almost connected to decreased birth rate as well. It's decreased by 45%. So the question is, are only Ukrainian nation now required some mental health protection? I would say probably no, because I'm not a person of statistics. Actually, I never thought I will be mentioned this in my presentations, but I just, before I start to prepare myself, and again, I would like to thank you for invitation, I start to learn what is, what about other world population? And actually, this is the live information. So when you come to this link, you can see in real numbers that even we have a lot of food, we have increased food production. But if you compare with deaths, diseases, everything could happen, because it's immediately changing a number. So we can see that we have a strange situation. We have a lot of overweight people, and we have a death from obese people, but we also have people who are dying from hunger today. And when I start to check what is the first place of death of people cases, I was actually a little bit surprised, because in my mind was that, of course, it's cancer, it's different AIDS, like infected people, or something like this. But I never realized that on the first place we have these communicable diseases, which are directly connected not only to stress, but let's say to our style of life. And also, I was shocked when I checked economical reports, and it was actually mentioned that we live now, and we will live a few next years, at least until 2025, what is next year, in the grip of austerity. So it's not actually very promising things. But even forget about statistics, we all know that we are working all the days, and actually we are faced very often to chronic fatigue syndrome, for example. And of course, it's known now that this chronic fatigue is actually connected to butyrate production. And this is, if it's butyrate production, that we know perfectly, so this is supposed to be connected to our microbiome data. And when I start to investigate more and more, and I realize that actually probably even the similar, if not the same mechanism between diseases actually and aging. And, you know, we know that we all prefer to have a healthy aging, but we know that there are a lot of disease associated with aging, like Alzheimer and Parkinson. And actually, if we would like to conclude a little bit here on this stage, that actually all these diseases like depression or anxiety, or even I was really surprised that irritable bowel syndrome, they have a similar roots, they have a similar mechanism. So now what is known for each one in this auditorium, I'm sure, because this is very specific auditorium, and I knew perfectly that these guys are, I mean, in our auditorium, it's very knowledgeable. So each one knew a lot about gut-brain axis. But for me, it's come like a new sign. Probably we can use these knowledges and especially take into account that depression and anxiety and different disorders also connected to hypothalamus, pituitary, adrenal cortex system. And with the same time, our fatigue and also age-relevant diseases and irritable bowel syndrome has a similar mechanism. Probably we can use these knowledges about gut-brain axis to apply for a, let's say, new treatment. And if you simply will take information from Wikipedia, so it's quite common to check first there. So you will find that anxiety disorders, and also, as I mentioned in the previous slide, major depressive disorders, myalgic encephalomyelitis, chronic fatigue syndrome, which I just mentioned, they all have, of course, they have a difference in dynamic and in different symptoms. And of course, they are different in the terms because, let's say, chronic fatigue syndrome is long-term illness where, for example, depression, it's 2 weeks. Or when we have an anxiety, actually, it's very difficult to tell how long it is because we are faced with this probably each simple day. But still, if we find all this, we start to understand that somehow it's connected, all these different type of diseases, it's connected to low-grade inflammation. And when I switched my interest to post-traumatic stress disorders, which is actually 1 of 2 of 20 individuals in the world are affected nowadays, I was quite surprised that all these symptoms like nightmare, traumatic memories, negative alteration in cognition, negative mood, depression, and anxiety, all these things are actually connected, because you can see in this paper and this slide, this is the first evidence reported by Israel scientists, and they found that all the symptoms are connected to oral microbiota signature. So what means oral microbiota signature? Of course, we all know that we have a different sites, mucosal sites, and these sites are covered with different type of microbes, and we call them vagina microbiome or sexual microbiome, or we call them gut microbiome, oral microbiome. But what I was surprised that, like signature, it means something more. It's molecular biomarkers, which you can really probably think how to use to detect the first symptoms. Because as you will read in this study, you can find it here, it was surprisingly that there are a lot of people, because it was a cohort study, which are investigated here, where underestimated. So they never even understand they have a PTSD syndrome, PTSD disease, post-traumatic stress disorders, because they never put in connection their psychopathology symptoms, all this anxiety, memory loss, and difference, with also idiopathic pain, with the connection of PTSD. So there are a few more interesting observations here, and I would like to draw your attention because, of course, the groups of investigated people here was veterans, but actually, if you read the conclusion, you will understand that also air pollution is positively correlating with PTSD symptoms. I guess it is now not a news, because we understand that in cities we have a huge population are suppressed with a different unpleasant effect and also unhealthy effect. And this is, of course, also connected to stress, increasing of stress in citizens from cities, big cities especially. And as you can see here, there are some specific bacteria which were decreased and correspondingly increased dependent of level of PTSD detected. And one of them quite interesting, anoxia. Of course, we need to understand that all these investigations are mainly local, because we know that everything like air pollution and even microbiome, it's quite a characteristic which are determined in majority by different local factors. So I put this specific bacteria, just if you will have a time and wish, you can check it on the web or whatever, and you can find that these bacteria are quite interesting because it's also predictor of parodontal diseases. So, and parodontal diseases, of course, I will not be probably able to discuss everything what is already known in this area, but this specific bacteria is very interesting because it connected to parodontal diseases, but in the same time, parodontal diseases connected to age-relevant disorders. So in my mind was like something specific what is actually were found in these 200 participants, and and actually thanks to this study, it was I'm using these slides instead of background and some sort of motivator, which pushed me to start to do some additional work in order to find, probably not in saliva, but probably also in gut microbiome, some signature, microbial signature, which we can use to detect early stages of PTSD. Because if these stages are detected on like early biomarkers, so we still have all the instruments, we still probably can correct it. So I just would like to mention one more time that it was studied about different scholars, and that's why important, because probably you will see a few slides where I will demonstrate that we come to also with a very multidisciplinary group, and mainly it was led by Ilana Gozet from Israel, and also it was a 4-decade-long study of veterans by Professor Solomon. And actually, I will probably now come to another background, which is known for you. It's also research background. It's about IPC Institute, and it's George Clark, which is known because actually all this gut-brain access, it's mainly developed by his team, and it's very good paper. Actually, if you can see the year, it's quite old paper, but in this paper you can read everything and you can understand that actually I start from conclusion of that paper, because it's very interesting, because the first of all, the paper is showing that we can regulate microbiome brain gut axis And if we can regulate this, but the issue is how we can regulate it and why we should regulate it, because we would like to get this homeostasis. And homeostasis, actually, it's some sort of physiological balance when God can play a role of early programming and later responsivity for the stress system appropriately, because it's like with the case with allergy, if it's not appropriate answer to response, we will have a disease. And actually, all this paper, if you read very careful, because it's actually about preclinical study and bacterial colonization of different type of mice. And all this paper actually is very important to let us know whether we can develop probably novel therapeutic for complex stress-related disorders. And another paper I also like so much, even I'm not a person who is very good in a neuroendocrine system, so you should forget me that probably not all the terms I will use very properly, but at least what I learned that it is a huge, actually it's published a little bit later, and what I really use and I appreciate that it still mentions that exactly the precise mechanism which are involved in this multi-directional communication between gut microbiota and neuroendocrine system still not clear. But what is clear actually? It's clear the way and all these regulators that neuroendocrine system actually connected directly, and it's mainly able to respond to stress with hypothalamic pituitary adrenal axis. So, and there are specific hormone, corticotropin releasing hormone and adrenocorticotropic hormone, and also I like this because it's clear for us that this communication, even we never will learn anything about stress, but we will know that HPA axis is connected to immune system, also to intestinal barrier, so it's permeability, a blood-brain barrier, microbial metabolites, because it's a main player, gut hormones, and sensory and autonomic nervous system. And actually, we understand that neuroendocrine system is classically defined as organized set cells for neural determination, which produce hormones or specific neuropeptides. And actually, hypothalamic pituitary adrenal system axis considered to be major neuroendocrine system, which is regulating different body processes, but we are mainly interested in response to physiological stress. And these hormones are known, it's not known hormones, but I would like to draw your attention mainly to glucocorticoids hormone. It's cortisol in human and corticosterone in rodents, so it's mainly hormones which are connected to stress. And this actually allows the authors of this paper to see whether, to detect whether increased activation of HPA excess can be connected to depression, but they check it, and yes, it is connected to depressed patient. But also, what is very interesting, this is connected since gut permeability to increase activity in irritable bowel syndrome as well, in a patient with irritable bowel syndrome as well. So if all this connected to cortisol levels and dysbiosis, it's somehow making possible to improve the situation. And to prove that it is possible, these authors are provided a different model of mice. I will not be able to describe all their study, but I would like to mention that the mainly all the factors which are acting, they are acting also in germ-free mice, but a less level. But so you can see that even germ-free mice, which are suppressed to some stress condition, they can regulate different, they can react to stress. So mainly they react to lowering of antibody production, gut function, T cells lowering, and they can increase or decrease a different type of metabolite profile like this. But to answer the issue whether microbial short chain fatty acids like, let's say, butyrate, propionate, acetate can play some role to induce gut hormone release, it was additional mice models used. So it's actually a review about preclinical study, and you can see here on the red, we probably mistakenly will be thinking that if it's red, so it means inhibition. No, by red you can see here stimulation. So all these things, GLP, prostaglandins, pro-inflammatory cytokines stimulate HPA axis, what means lead to the stress. And when we see on a green side, all this like GABA, leptin, they can stop. They can diminish HPA axis. But I guess you know this because GABA, it's typical what we can manage by probiotics and let's say improve our mood, let's say when we are eating a chocolate. Okay, so when we come to the conclusion here, there are some also signs, as I mentioned, microbial sign, that in Firmicutes and the Bacteroides representatives, they were like increased amount of Firmicutes and decreased amount of Bacteroides. It was typical for the patient with the irritable bowel syndrome, but actually the same was detected for depression, and also even more when was translocation made. So fecal transplantation study demonstrated if take transplantation of microbiota from depressed patients and to rodents, it will be the similar. It will induce depression-like behavior. So actually, even this was clear still in such a study, transplantation was induced immune activation and stress-induced corticosterone level were not affected. So there are still open questions whether it's connected to exactly to different hormones or not. But what is clear for us that at least 3 different pathways we need to pay attention, and this is very clearly demonstrated in this preclinical study. So there are such a communication between the gut microbiota and neuroendocrine system, like humoral pathway, because it's metabolites, hormones, immune pathway, because we have a induction of different pro-inflammatory cytokines, nerval pathway as well. Okay, and as I mentioned, we knew perfectly that there are some food or some bacteria like Lactobacillus brevis, Bifidobacterium dendium, they are able to produce gamma aminobutyric acid, which is GABA, which is really but the other bacteria species cannot, because they produce catecholamines. So we can mention this as a transmitters, and actually this is something what probably can be used. And this slide, which if you will ask my presentation, I guess you can get it because it is free for your using. You can see a continuation of this table. I cannot really move because it's open on all screen, but this is only the first part of table. You also can find this in published paper. And here you will see, based on different type of strains and bacteria, how they really influence on HPA axis and which type of pro-inflammatory or anti-inflammatory effects they can provide. And of course, reference who reported it. So there are a lot of study who is actually people who are trying to detect which type of probiotic actually we can use on, and which actually on animal species they were used. But, you know, the issue is when we are doing a lot of study on animals, and yes, we can really suggest that butyrated products in bacteria may control also appetite, because it's connected to mood, actually, because it is a food, and it's everything connected to cortisol and insulin, who is a little bit learn about hormone. And also, it's not only, I would like to draw your attention, it's not only lactobacillus, but also E. coli protein, caseinoleic proteinase B is antigen, which is mimic. So it's cross-reacting antigen with alpha-melanoxanine stimulating hormone. So there are a lot of data about bacteria, how they regulate, and different hormones. And also, in addition, it was shown that germ-free mice have lower levels of circulating leptin. That's actually probably they're more, let's say, more susceptible to anxiety. So here, in summarized, you can regulate, you can modulate, rather, you can manipulate with the gut microbiota if you know the way. But the issue is when it was done by different scientists, actually in different models, it was a quite strange conclusion made, which I can only mention that from one side, yes, we can use different strains, calculate different level of corticosterone like this. But when the different authors use oral antibiotics, or sometimes use a different strain of probiotics, unfortunately, they cannot find strong correlation or something which strain exactly can be used for a treatment, or at least even how the stress can be regulated, because there are quite complex system. Even it's clear that it's mainly the activation happen via immune system in response to stress. So now a little bit our history, which was mentioned. We are working in that field in the mice, a different type of germ-free mice versus conventional mice, and there our paper published in 2005-2006. And when we doing IBD model, why I come to IBD, because it's connected to irritable bowel syndrome, but it's immune bovine disease. And we also done some transferring of cells to SCID mice, which means several immunodeficient mice. We use a different subset of T cells. And actually, to demonstrate how IBD is happened, you need genetic factors, of course, but you also need enteric flora and dysregulated immune system. Why dysregulated immune system? Because we have a pro-inflammatory and anti-inflammatory, now it's very common pattern. And actually, we demonstrate that if you take a completely free mice from T cells and B cells and from bacteria as well, but then you add only one bacteria, Helicobacter muridarum, and also you add T cells, but which type of T cells? CD4 positive, CD45 RB high T cells, you will get inflammation only in that case. But then, of course, the mice which got this one bacteria, not conventional, and they become lost their weight because they have these cells, T cells, specific subset of T cells. But when you ask yourself why exactly, how it's happened, how regulation is happened, probably whether it's different type of T cells can be used, how it's connected to pattern, because we know that CD25 positive, negative, there are different T cells, because this is a T-regular T cells. When we sort all the different subset of cells, and also using germ-free model, conventional model, and mono-associated model with the transferred cells, it's a huge design here. I mentioned it's published, you can find it. So finally, you can see that only CD25 positive cells, it's here, and the blue color, can prevent IBD appearance. So actually, CD25 track T-regulatory cells can prevent IBD, which can be caused, let's say, if you can say T pathogenic cells with connection of specific bacteria. But also there are another mechanism, because we are speaking about a food. So it's not only bacteria, but also if you use the ovalbumin and you can prime the mice, and if these mice are primed before, but then they are fed with ovalbumin, you can also protect IBD by using this ovalbumin protein. So you can clearly see here that over-unfed mice and overfed mice, when the mice are primed and overfed, since the oral tolerance, they can prevent, they are not developing IBD. So if I will have more time, I will talk more about oral tolerance phenomenon, because this is actually connected to our answer to food. But if in general, if you use a different type of ovalbumin or conalbumin, and you have different type of the model, what we need to learn that unfed mice, unprimed mice, but finally germ-free mice, if you compare with conventional mice, they will never ever able to develop tolerization or to be tolerized for oral induction of a native conalbumin. But even if you use one simple bacteria like E. coli, of course it's not so big tolerance compared to conventional mice. But still, you can see here there are slightly registered difference. So it means even one bacteria, E. coli, can act like stimulate tolerization to different type of oral antigens. But when we come today to people era, and today era is not very simple, because the medicine is changed dramatically, because now it's social media, phenome, proteome, transcriptome, epigenetic, genomic, transcriptomic, everything. What is actually form a new medicine? A new medicine, it's, let's say, 3P medicine, 4P medicine. Actually, there are not so huge difference, because it's still evidence-based, but it's supposed to be predictive, so you cannot guess what will be happen with your patient if you will provide him with one or another probiotic, antibiotic, let's say, we will see. It's supposed to be preventive. Of course, it's supposed to be personalized and supposed to be patient-oriented. So you need to know a lot about patients. So you need at least to have some sort of individual profiles. And individual profiles, probably you need to know more about genome, microbiome, metabolome. And this is actually systemic approach. And for a first place, now belong these biomarkers, because you need to have evidence-based data to prove that this, what you measure in gut microbiome, in oral microbiome, it's real biomarker. So actually, this everything you can read about System Biology Institute. I like this institute because it's explaining a lot, and mainly about patient stratification. And also, as I mentioned before, and I would like to mention here, a lot of data you can find in Alimentary Pharmabiotics Center, which actually propose the term of pharmabiotics. And be very short, pharmabiotics, actually probiotics, symbiotics, immunobiotics, psychobiotics, whatever you want, biotics, but you need to prove it on clinical data. So it's actually not alternative medicine, it's just evidence-based alternative. So you need to prove this is working, then you can call it pharmabiotics. Okay, so we, at that time, we tried to face our research to obesity, diabetes type 2 patient, caries connected to enterocolitis with children, vagina microbiome, infertility, because it's now very common, and use depression. Use depression we did not finish. We started but did not finish, because, as I mentioned, it was interruption with the wartime. So it's still open subject, but I guess now it's become even more important than before. And also, which type of instruments we use? We use a different type of human dendritic cells models, mainly clinical trials, because, as I mentioned, mice fantastic, but you need to come to cohort study. Also, we done some in vivo study, but finally we come to, if this too complex subject, and how to calculate environment influence everything. So you need to create some databases. So you need to come to different type of information system, also geographic information system, because everything is local and so on. So another our history, I would say history 2013. We worked a lot, as was presented today by Slavi, that we worked a lot with a different type of project, and one of them was base food. It's Black Sea region food. Actually, the participants from Bulgaria were there. It is a Plovdiv University of Food Technology. I like it so much because we done a lot of work together. And actually, there are a publication about this. You also can read it. Actually, the site is still acting. There are a lot of data you can find there, even presentation. But what we actually find there, we found that there are a lot of more things which we can use an instrument to correct non-communicable diseases, which if you still probably remember now in the first place of this. So we use a different type of cells, we use a different type of commensal microbes, we use the microbes which we call postbiotics, which we isolated from edible plants and food, and we done a lot of work. It was a 3-year project, and we have a consortia from the different countries, so we are not only the people who done this work, but we have proposed a unique model and actually published with the Hungarian colleagues. And we were able to use these models for what? Just to not come into deeper details. But if you take individually from any person monocytes, so it's not really mature dendritic cells, immature, and you will make them maturation on a plate. And in these plates, you will add different type of bacteria. We use specifically, our interest was not lactobacillus, because we done a lot of work, lactobacillus, bifidobacteria, but we would like to see what happened if, what E. coli will do, because it's LPS producing bacteria, Bacillus subtilis, what we can get. And we use also pro-inflammatory, anti-inflammatory environment, because everything happened inside. And we put ATRA and without ATRA. And you can see here how differ the environment, how different environment acting on this different type of T cells, for example, or a different amount of interferon production. And we done a lot. We checked IL-17 production, T-regulatory cells, as I mentioned, CD25 positive, Fox XP3 positive, IL-10 plus T cells percentages. And we were surprised, because we are not expected the E. coli, even its commensal E. coli Schedler, will be coming on the first place. We thought at least it will be Bacillus, because it's very strong antigen. So finally, we were able to create a system with a different additional components and growing factors. And when we can really modulate, we can programming and reprogramming immune response. So actually using, by other words, the same bacteria, but using a different environment inside, we can do pro-inflammatory or anti-inflammatory effect on our organism. So this is only mentioned for us. This is a mechanism why it's come for inflammation, why it's protect inflammation, and this research was actually supported by a few grants. I would like to thank you for collaborators from Hungary, and this is an intergovernmental grants. But finally, what it tells us in conclusion, actually, that we cannot understand separately one bacteria or two bacteria or a few bacteria. We need to take everything, if you would like to be on a basis of a systematic medicine, so it's 3-pay medicine, 4-pay medicine, we need to check everything. We need to take into account environment, health indicator, nutrition, I don't know, everything. We need to create some sort of databases that we need to use the instrument for machine learning like this. So then we become the member to the people, Yetma, who created this paradigm. So we start to communicate with them actually, and we become an institutional member. You can find this. And then we start again our work. So we understand that in the classical medicine, you probably can go with observation treatment, not expected reaction. But here you can't. You need to have testing biomarkers. And this was published actually in 2010. And you need to understand which results you will expect. Then we start to use all our instruments from a cultivated bacteria, uncultivated bacteria, PCR, real-time PCR, biochemical diagnostic, everything, and also genome sequencing. It's 16 RNA sequencing. And we are using this now on a, I would say, on a daily basis, because we need to check, we need to prove which type of biomarkers we really can prove. And as I mentioned, 5 years ago, we were done a lot of experiments. So you can probably find this, our book, it's available in the internet, so it's Springer Nature. I don't have any rights anymore for that book, but it's here. For example, one of the paper, we clearly demonstrated, proved biomarkers which are typical for a different type of disease. So this is actually was our first step. Then we created some sort of IC instrument. I cannot change slides because it's actually on Ukrainians, but it's brand aliens, and we start to create some on a let's say on a clinical basis and the database is some sort of preparation which we call pharmabiotics, and we register them in pharmabiotics. And this is databases which are available, and even the name you can find. This is actually open access database. If anyone would like to get it, I can provide the link. This is about biological active compounds and their influence on different type of microbes. But the main issue was how to prioritize all these biomarkers, because you measure 62, sometimes even 100 different biomarkers, because it's social, psychological, and like this. So all this also published, so you can find our paper. It's open access paper. And so we found a new algorithm how to make them prioritized. So we measure them not like correlation, because correlation it's statistic, and we measure it's like in dynamic. Okay, so when we measure it's like dynamic, we found how not only bacteria, but how different biological active compounds can also influence differently on and can improve intestine microbiota and metabolize not only obese rats, but also we done experiments in people. And so we have some like another brand how to prepare these dishes, so we can share with you, because we are actually honored to tell that we learned the first, we created the first example of patient microbiome profile, but not a sick people. We use a like healthy cohort, but we detected in this healthy cohort, we detected atherosclerosis sign. Because, because before all the stress and anxiety or cardiovascular disease are coming, the first sign will be atherosclerosis. So it's our preparation look like this. So we can, we can do it. We have a lot of different patents also we can share, but I would like to draw your attention for the final part of my presentation. It's about atherosclerosis cohort. We use the 300 persons, Uzhhorod and Odessa, and it was started before war and it's finished after the war is started. So there are inclusion and exclusion criteria, and actually it was measured the pulse wave and the speed of propagation was measured, like elasticity of vessels was detected by non-invasive oscillometric device. Of course, it was not we have medical doctors for this. And this is actually how they can interpret, because they have specific formula. And here you can see two doctors who actually work with this vessels estimation. And with this actually machine, Dr. Taras Chandey Vasil Lohoida, PhD student. And actually we done all the biochemistry, immunology, inflammation markers, cytokines, etc., etc. So it's our team. Professor Volodymyr Artyomenko, he is from Odessa. And of course, we measure a lot of indices, as I mentioned. So the main issue was how to make them prioritized. We measure physical indices. There are a lot, and of course, it was something connected to weight, biochemistry, also known like glycosylated hemoglobin, LDL, HDL, and etc., etc. And we are doing also sequencing here. You can see a statistic. Not all the samples were good. But yet not all of the detected bacteria we were able to identify. But finally, we then to use all this pool of bacteria and use a PCA assay, as I mentioned. So it's principal component assay, but we use it for not statistical data, for dynamic. And this is actually results which we got from the library. It was looking like this. So all the data, sequence data, were sorted out. And as I mentioned, we have some classification results. And finally, based on all this study and analytics, we found that there are some, based on this cohort study, there are some specific bacteria were detected only in a group of people who has atherosclerosis sign. And some other group, so they were usually typical for a control group, but never have any sign for atherosclerosis. So then we are put also immune data, like different type of cytokines, and different other data mentioned. And finally, we found some mainly important bacteria, which with the high priority prioritization, we can probably think they can be used as biomarker for atherosclerosis. But when we keep doing our assay, we also find that, because I mentioned it's social workers as well, psychological, we found that general physiological condition and anxiety general is actually also very high. So they belong to 10 important parameters. So actually, by other words, we found connection between anxiety, because we have 3 different types of anxiety, and different type of bacteria and prioritization number. That's why we come to some sort of legislation with our documentation, and we start to use this, trying now to continue this study, if it's study, just to help our soldiers. And actually, this is also we are doing with with the estimation of T cells response. As I mentioned, you will not be surprising why. So we are measuring T cell response. And for what we are doing this, we just would like to prevent also, of course, as I mentioned, probably antibiotics, because antibiotics always playing a huge role in internal environment. So we are trying to put all this together, prevent multi-resistant strains, but also to choose specific bacterial cultures. Of course, we are working with lactobacillus because we can't really use E. coli or something because we don't have a documentation for them. But we are using an individual omics profile to choose not the strains which have no influence and not effective with those which can be effective against some specific strains. So we believe we are trying, because this is a very fresh photograph, so we are making such packets. We are trying to help to modulate their health, and we hope that all our teamwork, it's one team, and we have another multidisciplinary team, statistics, analytics, programming, and also you can find our contacts here, probably will be helpful to I don't know, to what we are doing, but also actually I believe it might be helpful for students for this course, at least in general. Thank you.
Thank you very much, Nadia. It was really interesting to see the complexity of the work that you're doing there. And now we open for questions. I have curiosity. Talking about these diseases like Alzheimer and other, do you think in the near future will be really a improve of the quality of life of these people because of the probiotics? Because in these days, we know that we cannot cure the disease, Alzheimer, but we can improve the quality. And do you believe that this is in near future or far future?
Thank you for the question. You know, actually, I'm happy that you presented me exactly that way, that if we are start to doing something, so we probably will get a positive result. And my answer will be yes. I actually were never ever interested in any elderly diseases like this, because probably, you know, you start to interested in when you become older, not before. But I was contacted with Dorofeev Andrei Eduardovich. You knew him because you met him in our meeting. You were invited speaker. And also another one, Oleg Shvets. We created the first in Ukraine. It's like memorandum of understanding, but a joint programming initiative. And actually, they were and are very ambitious. They would like to stop this age-relevant diseases. And that's why I start to contact with the people in dentistry, actually my department located in dentistry, because, you know, our problem probably, if it is a problem, we are microbiologists, and it's quite difficult for us to communicate with all these fancy doctors. And they usually mention they knew better than us. But you can see paper now that all the doctors become microbiologists. So now I'm honored to tell that, yes, in collaboration with them, if they will finally understand that need help us, I'm sure that we will be able, let's say, to cure periodontitis. Actually, it's not correct name. They call it periodontitis. That's it. So if we correct this, we can really prevent all this enterocolitis, all this toxication, and it's doable. And, you know, for me, since war started, you know, I was never ever thought I will be interested in PTSD at all, okay? Because we have a completely different things to investigate. But now, I guess, even our final study, when we are doing NGS sequencing, of course, we are not doing saliva because we don't have any database how to interpret saliva. But we have a databases how we can interpret the gut microbiome. So I guess now in my head, because working in USA, working in European countries, working in Ukraine, working in European projects, and come to my mind that everything is, of course, connected, it's clear. But if you will find the secret how to regulate this gut permeability, to stop all this process, we can do everything like a magic people.
Well,
so my answer is yes.
Like 2-3 years ago with Mike Chikindas, we had a paper on the probiotic from Bacillus. And when we was looking on the background on this area, it was amazing to see that actually not half, but maybe more than 30% actually of probiotic from Bacillus actually coming from Ukraine. And it's a very extensive work that is performed in this area of the world.
Yeah, but you know what is surprising? I also was thinking that people in the world, they will never ever accept it. And you know, one of the first probiotics which were validated by EFSA was based on Bacillus subtilis. It's almost killed myself because I thought they will never ever accept. Okay, but a lot of and it's great. Actually, it's very good. Why? Because Bacillus. I will not explain what means Bacillus, you know, but they are very strong. Sometimes I have what my huge interest now, you will be probably laughing from me, but E. coli, it's playing a huge role, it's like lactose positive, negative, all this different composition, okay? And, at the level, and it's playing a lot of in the gut. So, if you have some less amount than you need, you don't have a really healthy gut. So I was not able to demonstrate in my presentation, but we are never using a separate biomarkers. I guess not only we are using some sort of diagnostic ratio for different individual patients or people. Not all the people are patients, okay? So, and yes, and I guess, fortunately, so I guess it's very important to create something like, how to say, ecosystem between researchers. That's why I'm so happy to participate in this meeting. And I would be happy to attend another your meeting. And I'm thanking you so much that you are attending our meetings as well, because these meetings are mainly oriented in Ukraine on the doctors to provide them with new data. So I'm thank you so much for your question. And I hope probably if anyone will have some interest in our study, you always can contact me directly. So
and especially maybe later we can discuss the result that we had with Wilhelm Holzapfel and his, actually our students, Yuri Lee. We was investigating use of the bifidobacterium as an antidepressant, and there was some good results with them. But we can discuss in another occasion because we are actually presented now. So thank you very much again for your presentation. It was a pleasure to have you with us. Stay safe and
also my pleasure.
Near occasion we can talk again and you can share some of your great experience. Thank you very much.
Thank you.
So now we're going to have a short break, just a 1 or 2 minutes break, and then we're going to have a presentation from Professor Luis Augusto Nero, that he's let's say, starting from co-editor of the Brazilian Journal of Microbiology. He is the president of the organization of IFP Latin America that will be in a couple of months in November in São Paulo State in Santos. So if Nero is already online, maybe technical people, you can put it him. Boa tarde, professor Nero. Está me escutando?
Good afternoon, Slavi, and everyone.
Well, it's a pleasure to have the next presenter and to see him sitting in his office in far, far away place called Viçosa. And even the Faculty of Veterinary Sciences is even in the end of the university, so it's really far away. But well, why we have Nero today with us? Because Nero is a editor, co-editor of the Brazilian Journal of Microbiology, and he will give some information about this. And actually, maybe more important in this case is the fact that Brazil will be hosting the next issue of International Association for Food Protection Latin American Symposium that will be in November this year in Santos. And it's actually it's a quite an honor Brazil to be a host for this event, because it's not only Brazil. I mean, this event run all around in the Latin American countries, but there was organization committee was managed to convince the International Association for Food Protection that will be a good idea to have this in Brazil. So again, a little bit more information on this, and as well Brazilian Association for Food Protection that before was called ABRAPA, now it's changed the name to BRAFP. Okay, but Nero, please, you can give the information all about these events and the journal. And well, it's time for you to do the advertising job.
Okay, Slavi, thanks for the invitation. Thanks for the idea. In fact, we discussed that in the beginning of this week, and I have to appreciate that because it's a very good opportunity to share this information, some of the information that we are developing in Brazil and also in Latin America regarding food micro. So again, thanks Slavi for the idea and the invitation. Thanks Wellington for putting all this piece together in this very good initiative that you are having in this winter school. We don't have a very cold winter in Brazil, but we are in this season. Well, as Slav presented me, I'm the current vice president of the Brazilian Society of Microbiology, and also I'm the current president of the Brazilian Association for Food Protection. So the general idea that I would like to present you is just in a few slides, what is the current activities that we are doing in food micro in Brazil and also in Latin America in this context. And as the audience is all food microbiologists and people that work or study food micro, food safety, food quality, the idea is to present some good opportunities and alternatives for you to publish your manuscript, to attend some scientific events in the area, and that might be of interest of you. I will start talking first about the Brazilian Society of Microbiology, in which I am the current vice president. This society is a very old society in Brazil that has the mission to broadcast the microbiology knowledge to the common population, and the idea to interact researchers in different areas of microbiology. For sure, we have the food microbiology area in this society, in which Wellington and from USP and Virginia from Goiás Federal University, they are the representatives. We have a lot of activities related to microbiology in general in this society, including courses, lectures, events, and also we have the journal, the Brazilian Journal of Microbiology, in which the food microbiology section, now it's called the food safety and quality section, is a very important one with high quality papers and authors that always submit to us. We have some planned activities in this year and in the following year. Every 2 years we promote, we promote the Brazilian Congress of Microbiology. So in 2025, next year, most probably in October, we'll be organizing the Brazilian Congress of Microbiology, and we hope to have a very exciting and good news related to food micro in the following months. We are just setting up some agreements and some arrangements. We cannot say nothing right now, but the idea is to present in the next weeks or months how would be organized the food microbiology section in the 2025 Brazilian Congress of Microbiology. But in 2024, in this year, we are collaborating with the Portuguese Society of Microbiology to organize this International Congress on Portuguese Language of Microbiology. This Congress is an online conference. Will be held in between November 25 to 27, will be fully online. You can access by QR code the link of the web page of the Congress. And will be the idea is to promote some interactions between Portuguese-speaking researchers that work on microbiology. Food microbiology is one of the hot topics that will be discussed during this conference. So we'll have the participation of Brazilian and Portuguese researchers. The Congress will be online and fully in Portuguese. So it will be okay to attend and will not be so time demanding schedule. So will be very interesting. It's a very light Congress to participate. And the opportunity to present your abstract in your study. We'll select a lot of abstracts that will be presented online and will be some awards for them. You can submit your abstract until September 30. And another activity of the Brazilian Society of Microbiology, as I said, is the Brazilian Journal of Microbiology. This journal, we are promoting some updates in the organization of the journal. One of them was the organization in 6 sessions. So we have these sessions now, biotechnology industry, that there's some related with food micro, but it's very common to evaluate manuscripts with a very good interface with food micro in this session. Environmental and biodiversity, food safety and quality, that's the major one. Medicine and public health, soil and agriculture, and veterinary and animal production, that has some connection also with food microbiology. Luis Henrique Rosa and I, we are the current editors of this journal, and we have a very stable impact factor in the last years. We're keeping, even with all the modifications, with all the decreases that we're seeing in other journals, we're keeping the impact factor of 2.1. And we are, our current challenge is to reduce the time for the first decision. In the past, we were able to decrease from 2 months to 17 days. So we have a very good performance in this matter. So as soon as you send your manuscript, we are able to have a very fast analysis, and we're being able to publish very fast, very good scientific manuscripts. You can also access our website using this QR code. I noted that these slides are going down through the breaks of your school, so you can access all this information in their websites. And to finish this short and very brief presentation is just to present you the activities that we have for the Brazilian Association for Food Protection. So this association was started in 2002. I'm now the current president of this association, and it was the first one international association from Latin America that was affiliated to the International Association of Food Protection. You might know this association for sure. IFP is responsible for the publication of the Journal Food Protection, and also the responsible for the organization of the annual meeting of IFP that occurs every year in the US and eventually in Canada. It was on the major or the most important food safety and quality conference that we can attend, and everyone that were already in this conference know how it is good and the quality of the lectures. We have the focus on food safety and quality, and we target the collaboration among academia, industry, and also regulatory organs, always focusing on the food safety and quality to attend the consumers' expectations. As activities, we currently develop a lot of scientific meetings. We try to promote the scientific meetings at least once per year, but also we can advise in some consulting, and also we act as cooperation facilitators. It's very common when someone asks us or some company demands some expertise or some specific collaboration, and we try to find someone that might attend this demand from the food industry. We are being very successful in this matter. As Slavi already mentioned, we have an agenda for this year in 2024. We are organizing the Latin American event of AFP. AFP have this annual meeting every year, but every 2 years we have this Latin America satellite event that occurs in one country from Latin America. This year we are organizing this conference. Will be the AFP Latino 2024. So this conference will be placed in Santos, São Paulo, very close to São Paulo. You can just take a bus and go there. In 1 hour, you'll be there. And we will occur between November 11 to 14, just before the holiday that we have on November 15. So you have time to spend more days in the coast of São Paulo to enjoy your free time after the conference. We are organizing the conference with 8 keynote speakers and also 18 roundtables that most of you already proposed these roundtables, and we include in the program. We'll have 3 simultaneous sessions, and also we'll have the presentation of some abstracts that will be selected by the scientific committee until August or September. So if you want to attend this conference and present your scientific study, maybe in an oral session, and we will have an arena in which everyone that will be selected can present your selected abstract to the total audience of the conference. You can submit your abstract until next week on July 31. So you have 1 week, in fact, to finish the submissions. And we have a lot of awards. You can check all the awards that we are offering to the presenters in our webpage. We already have 4 distinct awards from the Brazilian Association for Food Protection, the ICFMH, FEMS, and also the Brazilian Society of Microbiology. So it's a good opportunity for you to present the results of your research, to interact with different researchers from different countries, not only Latin America, a lot of other countries researchers from other countries will be attending, and to also present your results. So this is that I want to present, and Slavi, if you want to ask some questions, I'm open for doubts and questions that you might have.
Actually, can you hear me there?
Yes, for sure.
Okay, so give him applause first. And now I have a few questions. First question.
Yes.
You mentioned that the deadline for the IFP Latin America is now on 31st of July.
Yes, next week.
Any chance for extension?
We already extended one.
So no
originally it was June 30. We are expecting about 200 abstracts. We are almost reaching that. If you reach this target by this weekend, most probably will not extend, but depending on demand, we can extend, but for sure, this will depend on demand. So if you have some important study that's about to finish and you have 1 or 2 weeks more to finish and to organize your abstract,
but better don't count on the extension. Better submit in the next 10 days.
Yes.
Because maybe you can lose the opportunity.
Yes. It's been very difficult because as much as we have enough time, we know about that, we are aware about that, but as much as we delay to close the submissions, we will have a lot of abstracts. For sure, this is good, but we have a limited space to present that. So we need to manage the presentations properly. So if you have some abstract,
I know very well when the IFP in the US or Canada, they organize, they never give the extension.
Oh, yes,
we
they
need
never.
to respect these deadlines. Yes. I mean, they're not Latin Americans. They have the real deadlines and they need to be respected. Regarding the Brazilian Journal of Microbiology,
yes,
I think something that you need to mention, because I think it's important, if I'm not wrong, you don't charge publication fee to publish in Brazilian Journal.
No, we don't have publication fees unless you want to publish open access.
Yes, so it's a hybrid form.
Yes, it's a hybrid form. If you want to, yes, for sure you have to transfer the copyrights to Springer once accepted, but this is common to every journal. But if you don't want to publish as an open access that everyone will be to have access on that, we don't have to charge the publication fees for that.
Yeah, it's important because everyone knows the financial situation, so we cannot spend too much money on publication. So
yes, I understand and I agree with you. If I'm not wrong, I think Brazilian Journal of Micro and Springer, they are on the Periódicos Capes. So for us in Brazil, it's not so difficult to reach and to access the manuscripts on the Brazilian Journal of Micro.
And you mentioned that impact factor, it's 2.21.
2.1, I think, yes.
And which Qualis 1, 2? Not sure.
I'm not sure about the Qualis, Lav, I have to say to you, because depending on the area, they fall in different ones. I know that they are trying to unify the Qualis now, and I know that they are trying to have a discussion on the Qualis for the next evaluation. But what I know is that the Brazilian Journal of Micro is very high qualified, based on international standards. We jumped from the microbiology area from the quartile 3 to 2 in the last analysis. That was a very good indicative. So,
by the way, just to share something, my third article in my life actually was published in Brazilian Journal of Micro.
Oh, really?
Congrats. And I wanted to be published there. At that time, Bernadette Franco was the editor of the journal.
Good.
Well, I want it and I have it.
You can submit another one.
Well, you will think about.
Yes.
Well, from the audience, questions. So just let me give the microphone. We have Emilia Lima, a postdoc from the São Paulo University.
Good.
Hi, Professor. About the BRAFP, could you tell us about the importance or the benefits of being a member of BRAFP?
Thanks for the question. This is very important. First, because what made the society is the members of the society. So as much as we have members, more we can develop some activities to improve the interactions and also the cooperation among the researchers. As a student or as a professional, the first point that I need to mention that the membership fee that we ask for the one that would be interested to join BRAFP is merely symbolic most of the time. It's not too expensive. I think the fee per year is 100 or 200 reais. It's very low compared to other international societies. And the good things, the good thing that being part of scientific society, that will be part of these activities that we can organize. All members of BRAFP have discount in these events that we are partners. So we always try to collaborate with the Brazilian Society of Micro, with other Latin American societies. And when we have some event or some conference, we have discounts to register in such events. If you take a look in the registration fees for the IFP Latino that you say, you noted that for the members, the BRAFP members, we have some discounts. So this is of interest to everyone that might be joining us. And also we have some opportunities. From time to time, we have some corporations that they give opportunities for the members of the society. So for courses, for some newsletters with some specific opportunities. So this would be a very good point to start to interact scientifically with other researchers in the same area. We still don't have any scientific journal. We don't have enough funds for that. But I think, in my opinion, I think the activities that we're trying to push everyone to organize and to organize some scientific events and to give discounts for the members is very, I think it is more than enough to cover the investment of becoming a member of the society.
Thank you very much. More question from the audience? I have a question to the audience. Who will be attending the conference in Santos? 1, 2, 3, 4, 5, 6. Well, you're having quite a big fan group from here. Okay.
You still have time to submit your abstracts and go there.
Well, they already have it. Most of them they already submitted and been approved. Okay. If we don't have more questions from the audience, there's no one asking from the... no, there is a question. Oh. Where will be the next Congress of Brazilian Society for Microbiology? Wellington Manuel Pinto.
I know him.
He's a very brilliant guy.
We are discussing that. We are most probably will be in the Northeast region of Brazil, because we always had the last ones in South. And we are trying to find a good place for that. Most probably we will divulge that maybe in November or even before that, but in November we have some plans to have some official announcements during the FP Latino in Santos.
Well, maybe it's a good idea to announce this in IFP in Santos, as you mentioned. Could be.
Maybe, yes, because we have another announcements to do that, but we just need some time to confirm these announcements.
Okay. So thank you very much for your participation today. Enjoy the most probably warm weather in Viçosa.
Yes.
And hope to see you soon in another occasion. Thank you very much. And we're going to a short commercial break.
Okay. Thank you. Bye-bye.